moment. Halo, diceklik.
Yeah. Okay.
Layarnya jenengan terbalik, background.
Oh, really?
Ya, backgroundnya jenengan masih terbalik. Halo, Prof
Something like
Rosita.
this?
Hai Bu Uun, apa kabar?
Tambah glowing ya. Wow, glowing sekarang.
Bab kerjanya cuma makan.
Is it okay now? Is it okay?
Bu Umi, bisa dibetulkan backgroundnya Bu Umi? Masih terbalik terlihatnya.
Something like
FPIK.
this? No?
Ya, virtual backgroundnya terbalik.
Really?
Iya.
Mungkin karena di mirror, Bu.
Iya, mirrornya diceklik.
Coba di uncheck aja, Bu, mirror.
Sebentar. Kalau di uncheck seperti ini, tambah enggak?
Dari kemarin, Bu, itu kebalik.
Yang mirror diceklik. Mirrornya diceklik.
Lagi, Bu. Nanti oke. For a
Atau
while,
nggak apa-apa.
saya tak pakai background.
Saya sambil ngajar, Bu.
Nggak apa-apa, wis. Oke. Nggih. Matur sembah nuwun ya, Budin. Saya sambil jalan ini ya.
Sambil ngajar.
Oke.
Sorry, ya. Probably okay,
Bisa
later I will
dimulai,
change.
Bu Umi.
We are just about to start. Ya, terima kasih. Thank you, Bu Uun. Good morning, everyone, dear colleague, dear participant. Hope all of you are in a day. Now, the second day for us to listen to the lecture of Prof Rosita. And the topic of today is about some supplementations. Yeah, maybe I can.
Agak putus-putus suaranya, saya enggak seberapa dengar.
Responses.
Okay.
Halo, Bu Umi.
Share screen for today. Actually, yesterday was today's topic, but since there's some changes, so yesterday was only like introduction. So today we will have the first topic, the real first topic, molecular immunology development of genetic cultivations, the effect of supplementations in immune responses. Before we start, Prof Rosita, probably I will just say the schedules today, so that we already commit from the beginning. For instance, like the first lecture will be given like half an hour, one hour and a half first, and then there will be a pause a bit for question and answer, and then we will continue again for the second session for 1.5 hours, and then probably 1 hour, and then question and session again, question and answer again, and then the third part you will get the last part of your presentation, and then followed by the last question and answer. So probably our schedule today is like that. What would you say, Prof? Is it okay for you?
Yeah, it's okay. I think we will stop in between what you suggested, like 1 hour or 1.5 hour like that. We stop whatever you want to do. You want to grab a coffee or want to go to washroom and question and answer, and we continue again.
Yeah.
Yeah.
Yeah, so you also have sometimes to take breath, not directly for 3 hours, 4 hours lecture will be too much.
Yeah.
Okay, dear participants, we will begin in shortly. Please take advantage of what Professor Rosita will share with you, so that it will give you what is that, an opportunity for the future. Okay, Prof, please, time is yours.
Thank you, Bu Umi. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning, respected Dr. Oon, Bu Umi, and if there is lecturers here, nice meeting you again, and of course our beloved students. Okay, I will continue the second lecture today on a topic given to me on effect of supplementation on immune system. I will try to share. Have you stopped sharing, Bumi? I think you have. Okay. Can you see this? Sorry. This is not it.
Yes.
Can you see this one?
Yes.
Yes.
Okay.
Yeah.
All right. Okay. Hang on, yeah.
Kuya berukun, diklik deh kuya berukun. Nggak salah di share Mbak Septi.
So we have covered a very fundamental topic yesterday about physiology, about the organs, about the process involved in a fish or even in a crustacean, in a shrimp. So today we're going to see the effects of supplementation on immune responses. But before we be able to see the effect, then we have to have some knowledge. I know you have covered maybe in your degree or in your S1, S1, you have covered the immune system subject maybe, but this will be a bit of revision again. Okay. As we all know that the main purpose of aquaculture farms are to obtain optimum growth in reduced time. We, as much as possible, we want our fish, our shrimp to grow with good growth in a very shortest time possible, because after all, when you are venturing into aquaculture, at the end of the day, what you want is a profitable venture, a profitable business. So we want our fish, or even your shrimp or whatever you are farming, seaweed, you want they grow fast in a very possible shortest time. So that is the number one. Number two, apart from that, you want to maintain also good health of your farm species, because we know that diseased fish, diseased shrimp will not produce good product. You know, it will affect your price, and even worse, you won't be able to sell them. Yeah, if your stock is collapsed before the harvest period. And then the other purpose of aquaculture, of course, even though we said just now we want to produce fish with very, very good growth in the shortest time possible, we, at the end of the day, would like to maintain the good quality. The final product must have good quality, especially when you're dealing with export of your fish product, because there is certain standard, certain guideline that you need to meet before you can sell your product. Yeah, but all these three must be obtained, must be implemented through sustainable aquaculture practices. which I will cover on Thursday. Okay. Okay, let's have a look at how fish stress and health affect our the fish immune system. Fish are usually cultured in enclosed spaces. You know, we use tank, we sometimes use pond or in cages, and this captivity will somehow, like it or not, will increase will put some stress to your fish, yeah, because fish in their natural habitat, they are free to swim, not unlike in the tank or in the pond or in the cages. They have restricted movement. So like it or not, aquaculture actually, especially at the initial of the culture period, will put the fish in stressful condition until they are already adjusted into the system. Maybe they will be better after that. Okay, like what we said, the purpose of aquaculture is to get maximum growth, to get profit. So we tend to stock our fish at high density. So overcrowding will adversely affect the fish health. Yeah, because we want to produce high volume. So we tend to stock our fish very high density, for example, in a cage. So this is also a situation where fish will be exposed or will be affected in terms of health. The culture environment also tends to produce poor physiological condition for fish and increase susceptibility to infection. Culture environment means, you know, the water quality, maybe there is pollution, there are predators in the nearby cage. This will put under stress, yeah. However, the use of chemicals and drugs in aquaculture system will cause a residual problem and poor quality fish and its product, because like 10 years ago or even 5 years ago, when we have issue with disease, we always treat them with chemicals and drugs, yeah, which cause problem for a long-term use of antibiotics and all that will have issue in our final product. And of course, vaccination are also applies in aquaculture industry, but vaccination is not so straightforward. Normally, they are not able to confer protection on their own, especially those vaccines based on recombinant antigens and or inactivated pathogens. So there are many issues, but I know there are also some success story about using vaccines in aquaculture. So the use of alternative, preferably not expensive and effective product, a natural product as supplement to boost immune system is highly recommended these days. Yeah, we are going going to practice the green technology, meaning the use of natural product instead of drugs or chemicals. Okay, let's have a look at stress. The general physiological response of fish to threatening situation, as with all vertebrates, including like human, is referred to as stress. Yeah, meaning to say, when we are exposed to a threatening situation, we will feel stress. Same thing goes to fish. A stress response is initiated almost immediately following the perception of a stressor, meaning our body will act immediately after we are exposed to a stressor, whatever stressor, in this case, the fish, rough handling or high temperature, whatever stresses. So immediately the body will respond to this stressor. Mildly stressful situation can have beneficial or positive effects. They call it eustress. While higher severities induce adaptive responses, but also can have maladaptive or negative consequences. So in other words, not all stress is bad. If it's only mild, then maybe it have some beneficial effect, yeah, because it can induce this adaptation in the body, but not the high stress condition. The stress response is initiated and controlled by two hormonal systems, those leading to the production of cortisol steroid, mainly cortisol. So if you notice, most research, when they want to study about stress response in fish, the parameter that they will determine, one of them is cortisol, because cortisol is an indicator, yeah, indicator of stress. And catecholamines, this one is such as adrenaline or noradrenaline and their precursor dopamine. So together these regulate the secondary stress response factors that alter the distribution of necessary resources such as energy sources and oxygen to vital areas of the body, as well as compromise hydromineral imbalance in the immune system. This is just an introduction. What is the stress all about? I am sure everyone in this Zoom have seen this diagram, correct? I don't think anyone never see this. This is in our very maybe your I don't know what you call a secondary school, the relationship between host, environment, and pathogen, and this one, the disease. Yeah, this is just a revision for you to remember. Okay, this one also another revision. Fish diseases can be divided into several types. One is the one caused by bacteria. These are the example: red paste mouth fungus, tuberculosis, dropsy, banyak lah. Okay, fungal, argillus, ichthyoporidium, parasitic. Yeah, I will show you the example later on. Protozoan also another type of disease. Non-infectious. Okay, in general, disease can be divided to infectious and non-infectious. This can be infectious. This one non-infectious. Tumor, congenital abnormalities, normally genetic origin, injuries, constipation, got nothing to do with the the pathogen. This is based on other causes like malnutrition, nutritional based disease, deficient in certain nutrient, for example, vitamins or mineral can cause also abnormality. Or this one is another type of disease, miscellaneous, that doesn't belong anywhere. This eye disease, swim bladder disease, and of course, another important disease is caused by virus. This is example of different diseases. This one we took it from our own hatchery. Yeah, we have Garupa, hybrid Garupa, I think. This is heavily infested by marine leech. Yeah, because this marine leech is very difficult to remove from our system, from our tank. Because they have dormant seeds sometime, after a condition is optimum, and then they will hatch out. So it's very difficult to totally remove the leech from the system. Sometimes you have to do a total cleanup whereby you dry your tank, everything, and then disinfect with some chemicals or whatsoever, or chlorine. Even that also later you still can have this outbreak of parasite. This is example of fish infected with bacteria, also a grouper. This is koi infected with herpes virus. Yeah, the operculum heavily affected already, rot. Yeah. This is typical fungal infection. White patches will be clearly seen outside the body or sometime inside the gill. This is the protozoa. In this case, is the Ichthyobodo. I'm sure you remember your fish and health subject. This is the example of disease that doesn't belong to the cause of the pathogen. Normally, eye disease, you know, accidentally the fish rub the eye into some rough surface and the eye become cloudy. So it also another type of disease. Okay, I just want you to see, this is the viral disease video.
My name is Nick Phelps. I am the aquaculture specialist at the diagnostic lab. My responsibilities are to oversee the fish health testing at the university. Well, the VHS was just... My name is Nick Phelps. I am the aquaculture specialist at the my responsibilities are to oversee the fish health testing at the university. Well, the VHS was just found in mid-February by the Apostle Islands and Lake Superior. So it's the first time that in the furthest west it's been isolated. So it just shows us that it's continued to move throughout the Great Lakes area. There's been some speculation that the virus has kind of settled down and hasn't moved very much in the last couple of years, but that just isn't the case. Although we aren't seeing these prolific mass mortality events, the virus is persisting, and that's real important information. It's important for the bait fish industry, the harvesters and growers. It's important for fishermen to know that this disease is out there. What they can do to prevent the spread are preventing fish from moving from one lake to another. That includes bait fish, fish that they have in their live wells. Water can transmit, the virus can live within water. It can live on the side of a boat, in any of your nets, anything that's wet. It can potentially transmit the disease. So like the same precautions that you would hear about milfoil and the zebra mussels can be said for viral hemorrhagic septicemia. With that many fish moving around and this disease on our doorstep, we need to know where it is as soon as it shows up. It's important for anybody interested in the health of Minnesota lakes to prevent another exotic species, in this case a disease, from emerging here.
Okay, so as you can see, hang on, yeah, I don't know how to do this. Okay, as you can see, that is one of the incidents of viral disease. So precaution and management is important. How to handle or prevent the viral disease from spreading, yeah, in terms of your what do you call in this case in the video they stop the movement from fish from one lake to another lake or something, you know, not using the same equipment, something like that to prevent the viral, because viral disease normally is one of the type of disease that is very little for your farms, you know, because it will immediately kill most of the biomass in your cage or in your pond. Okay, so now we come into the immune response system. Okay, if you can see in this diagram, this is a comparison among the more primitive fish to the cartilaginous is the elasmobranch, bony fish, amphibian, bird, and even us, mammals. So if you can see, there is no information in Agnetha here. And then this cartilaginous is the elasmobranch. In this case, the example here is the shark. If you see, they have this epigonal. These are the name of the, what do you call, immune system of shark, yeah, which is different from teleos fish, yeah. Okay, same thing with leydig also. Thymus can be seen on other fish, and same thing with what do you call, same thing with spleen, yeah. Okay, if you later we will discuss more on this organ, but not so much on shark, because we are focusing on aquaculture species. Okay, in bony fish, the teleos, we have head, kidney, thymus, and also spleen. So the same thing, but in amphibians start developing bone marrow, which is not seen in aquatic animal, these two here. Yeah, but in amphibian, we can see this bone marrow already, and then even the terrestrial animal, they are more complicated immune system already. And then, of course, the most developed immune system is in human. This is just to compare what is the immune system like across the animals, from the aquatic to the terrestrial animals. Some are unique for that species alone. For example, epigonal, leydig are unique to shark. Okay. In principle, immune system fish equals that of other vertebrates. Yeah, not so much different. However, there are differences, especially in histopathology and location of important immune competent cells. The function of the fish immune system are equivalent to those in other vertebrate. Function is still the same, which is to against disease and to protect the cells. Fish comprise the largest vertebrate class, very diverse in evolutionary terms, and may be divided into jawless fish and jawed fish. So jawed fish will be further divided into cartilaginous and also the bony fishes. So we will strictly focus on the modern teleos that are commonly cultured in our time. This is just to give you some background on the immune response system of different animal. Okay, I'm sure you have covered immunology maybe in your undergraduate degree. For those doing master now, are we having a bachelor degree also today, Bu Umi? Is this
Yes.
only master student, undergraduate also?
Yes,
Yeah.
undergraduate.
Oh, okay. All right.
You can see in the participant name when
Okay.
S1 means undergraduate, S2 is postgraduate.
Okay.
Professor.
All right. Thank you.
Yeah, you're
So,
welcome.
oh, sorry. Okay. So, fish immune response system. I know you have maybe covered this topic in your early semester. So, immune system has developed a broad area of defensive mechanism to protect the host from a high variety of pathogenic organisms and toxic substances. Yeah, so not only immune system is not only to fight disease per se, but of course the stressor. You remember the word in the stress slide just now? Any form of stress can cause or can reduce our immune response. So toxic substances also is another stressor. So in general, fish immune system is categorized into innate and adaptive. These two keywords here you need to remember, innate and adaptive immunity. Innate, some other terms are also used like non-specific, or is genetic immunity, meaning immunity of an organism that they have when they are born, or in this case when they were spawned. Yeah, immunity an organism is born with, that is innate immunity, or non-specific immunity. This is determined genetically. Yeah, you're born with it. And the other one is adaptive or specific acquired immunity. This is the immunity that an organism developed during the lifetime. Slowly it developed, during the lifetime, not the one that they have when they were born. It's not genetically determined. So these are the main categories of fish immune system: innate and adaptive, or specific and non-specific. Okay, let's have a look at the organs that very important for immune response. Thymus. Thymus together with the kidney at the anterior and posterior and spleen are the largest lymphoid organs in teleost fish. Yeah. So that's why I start with fish physiology yesterday, just to give you a better memory. What is the organ that is responsible for certain function, or yes, for different function. Thymic structure, contrary to higher vertebrate, is highly variable, and in many species it's not possible to clearly differentiate between the cortex and the medulla. So thymus will not be the same in different species. Yeah, they have different size, they have different structure depending on different species. Kidney. Kidney in teleost is equivalent to bone marrow in vertebrate. You remember in the diagram just now, only starting from amphibians, and then terrestrial animal, they started having bone marrow, but there is no bone marrow in fish. But fish has kidney, which also equivalent, the function equivalent to bone marrow in vertebrate. So this is actually the largest site of hematopoiesis until adulthood. Hematopoiesis, I think everything starts with hema, it's related to blood production. So kidney is actually an important organ in fish. The main cells found in the interior kidney are macrophages, which aggregate into so-called melanomacrophage centers and lymphoid cells, which are found at all developmental stage, mainly B cells. Okay, another important organ that is related to the immune system of a fish is the spleen. Spleen is distinctly divided into white and red pulps, even if structurally less organized. This dividing is very variable. In some species, the red pulp prevails and may include the whole organs, whereas in other may be composed only the lymphoid cells and macrophages. Fish spleen is the main site of erythropoiesis, phagocytosis, and antibody formation. In bony fishes, it is generally accepted as many secondary lymphoid organ, which a plenty of B cells are activated and differentiate into plasma site. So it is the second organ after the thymus. Yeah, it's not the first lymphoid organ. It's still important. So when you read papers on immune system, you will come across with this term, immunoglobulin. Always you will see IgM. What is IgM? So if you read publication, journal, or book, you will definitely come across with immunoglobulins, because immunoglobulin is very important part of immune system. It is a class of proteins actually present in the serum and cells of immune system which function as antibodies. So if you do testing, recently I've done my antibody test, not antibody, yeah, antibody test for COVID. I have done my second dose, everything. I just want to make sure that I have the COVID antibodies. So I went to a test, then, you know, this is immunoglobulin method that they show me that I have this antibody in my blood. It's a very straightforward test. Just they prick your blood from the finger, and then it's just a test kit. Yeah, it's not a lab-based test or analysis. So this is immunoglobulin. Yeah, the first antibody the body makes when it fights a new infection. Immunoglobulins of fish are found in the skin. Interesting, right? Fish skin has antibody, gut, gill, mucus. You remember someone asked me yesterday how gills protects or become involved in immune system of the fish because of the mucus, and the mucus contain antibody, immunoglobulins. So I think that was a very good question yesterday that you asked how gills involved in the immune system of a fish, and then bile and systematically in the blood plasma. Okay, let's have a look at this fish organ. This is kidney. So kidney, they have several parts. What they call here head kidney, this is the posterior part of the kidney, is actually the central organ for immune, endocrine interaction, and phagocytosis. And then this head kidney involved in development of B lymphoid cells. This is just the name of the cell. I don't know why they call it B or whatsoever, but this is the name of the cell that involved in immune system. And then the formation of immunoglobulin and immune memory. So kidney is quite important in fish as an organ to defend the pathogens or any other stressor. Okay, thymus. This is the thymus, is an organ for phagocytosis. Produce T lymphocyte involved in stimulation of phagocytosis, stimulate antibody production. And then spleen. Spleen acts as scavenging organ, although we said in the earlier slide, this is like a secondary organ, play a vital role in hematopoiesis, antigen degradation or trapping, and of course, antibody production. What is this? I can't really see. The main difference between T cell and B cell is that T cell can only recognize viral antigens outside the infected cell, whereas B cell can recognize the surface antigen of bacteria and viruses. So this is just to differentiate what is the difference between the B cell found in head kidney and the T cell found in thymus. So, okay. Okay, what is immune response? Immune response is the reaction of the cells or fluids of the body to the presence of a foreign substance. Yeah. So, for example, here, immunomodulatory nutrient and additives, they are taking nutrient, additive, supplement, and then other stressor, pathogen. Remember, not all stressor is in the form of pathogen. It could be in the form of toxin or anything that is not pathogen related. And then the activation of immune system and the other process involved, antioxidant defense, cellular responses, the nutrient release, cytokine, acute phase protein synthesis. So this is the outcome, restoration of homeostasis and hostile milieu for pathogen. Okay, let's have a look in detail what is the innate or non-specific or genetic immunity. The fish possess a well-developed non-specific immune system, yeah, not the specific one, but they still have specific immune system, of course. The innate immune system is the first to respond to pathogen and does not retain memory of previous responses. So meaning the innate immune system, when fish have attacked by or infected by pathogen, innate immunity will do the job first. But after they do the job, they don't remember. They don't retain the memory. So what are the organs involved in the first line? The skin. Of course, skin is the first organ will have contact to the water, to the environment. Lateral line, like I said yesterday. Gills are the first line of defense against pathogens. The skin of fish is coated by mucus, which is continuously secreted by goblet cells and contain immunoglobulin, right? Antibodies as well as lysozyme. The components of the innate immunity are divided into humoral molecules that are freely located in body fluids and cellular components. These include the growth inhibitors, lytic enzyme, agglutinin, precipitins, cytokines, chemokines, and bacterial peptides. There are many different components. So innate immune response involves a large number of cells and molecules. There are many cells involved. Thus constitutes the first host response. Yeah, immune system, innate immune cell include basophil, other dendritic cell, eosinophil, monocyte, macrophages, neutrophil, and natural killer cell. So these are the name of the cell involved in the innate immunity system. Okay, let's have a look at adaptive or specific immunity. If a pathogen persists, the adaptive immune system is recruited. Meaning, kalau if the pathogen is infecting for long term, then the system is activated. The adaptive one is activated. The adaptive immune system is highly specific to a particular antigen and can provide long-lasting immunity. So this is contradict to the innate immunity or the non-specific immunity, whereby the innate immunity, they don't store the memory. They don't remember this antigen, what are the pathogen attacking. So they don't store that memory, but adaptive immunity will have long-lasting immunity. This is like what we are taking now, the COVID vaccine. That is the adaptive immunity, right? We have the vaccine, which is very specific to target certain antigen only. Having a COVID-19 vaccine will not give us protection to other types of virus, only the COVID-19. Even COVID-19 also mutate to other strain, which maybe our vaccine may not be as effective as to combat the real antigen that is developed for the vaccine. Same concept apply to this one. So central to the adaptive response are the lymphocyte, cell types responsible for the diversity of antigen recognition, specificity, and memory. Adaptive immune response involves a few number of cells, and it is based on the antigen-specific receptor expressed on adaptive immune cell T and B lymphocytes. Okay. When is the adaptive immune system activated? Like I said just now, it is activated when pathogen evades the innate immune system for long enough to generate a threshold level of an antigen. Yeah? You remember the first defense is the innate immunity, but if it's invade for long, then only this adaptive immunity is triggered. And antigen is any molecule that induces an immune response, such as toxin, is a molecular component of pathogen cell membrane, is and unique to each species of pathogen. So this is what I explained about. The example is the COVID. Yeah. Okay. There is some differences between fish and shrimp when it comes to immune system. As you can see in this diagram, fish is vertebrate, shrimp is invertebrate. So the defense mechanism of invertebrates, especially in this case, the shrimp or crustacean, is less developed than the finfish or other vertebrate. Specifically, crustacean have no adaptive memory. Yeah. In other words, they do not have the ability to produce immunoglobulin, so that they apparently depend only on the non-specific, the innate defense system. Unlike the fish, at least they do have adaptive memory. So let's have a look at the immune system of invertebrates or the crustacean. This will be mostly on the commonly cultured species like the lobster or crayfish, crab, shrimp. So in this species, the physical barriers are the first obstacle to detain pathogenic microorganism. If you notice fish and crustacean, they have different morphology, right? I think everything is created in a very brilliant way. Since fish have both non-specific and specific immunity, hardly you see fish with this carapace, right? Only crustacean have those armor, the carapace outside their body, because they don't have a specific immunity. They don't have adaptive immunity. They are depends 100% on their non-specific immunity. So the first physical barrier is the most important part to detain or to what do you call, defend themselves from pathogen or other stressor. That's why they are created with this special covering if you compare to the fish, because they don't have that adaptive immunity if they were to be attacked later on. So that everything created in this world is so unique and with a purpose. I always believe that nothing is created for nothing. I truly believe in that. tidak ada yang sia-sia yang Tuhan ciptakan, because it always has their own value, their own use. Just take this as an example. Fish and crustacean. Fish morphology doesn't have this unique carapace. They don't have the outside shell. Why? It is explained by science, right? So it is very interesting when we explore other creature in the world, that we understand the purpose. You know, everything is created with purpose. So when there is damage and the microorganism invade the tissue, proteolytic pathways, everything to do with the protein breakdown. You remember yesterday we were talking about proteolytic analysis, what happened in digestive system. So this pathway take place instantly, allowing elimination or diminution of microbes invading the organism. The effective mechanism for integrate immune responses include the coagulation cascade, which avoids the loss of hemolymph and stimulates oxidative metabolites and production of melanin by activating the PROPO system. I explained to you what is PROPO. I use the short term here. This is PROPO, yeah, prophenol oxidase is kind of enzyme, yeah, prophenol oxidase. Yeah, we use PROPO as a short term. So let's have a look at immune system of invertebrates. Crustacean possess an open circulatory system where nutrient, oxygen, hormone, and cell distributed in the hemolymph. So the hemocyte, the circulating cell, could be functionally analogous to vertebrate leukocyte because they are mainly involved in self-matter recognition and elimination, as well as downstream coagulation. So a crustacean possesses several defense mechanisms that become activated depending on the pathogen attacking them. So when foreign material is recognized, different effector mechanisms are activated, such as this PROPO system, or even phagocytosis and encapsulation. So these are the example of how crustaceans defend themselves from pathogen. This is just a revision only, just in case you don't remember what is hemocytes, what is phagocytes. So hemocytes are cells that possess functional devices such as macrophages, granulites, and natural killer cell in vertebrate animal. Hemocytes play an important role in the immune system of crustacean body by expelling foreign particles through phagocytosis, encapsulation, and nodular aggregation. So the marker parameters for the evaluation of shrimp defense system involve humoral and cellular immunity, which include hemocyte count, PROPO enzyme, and phenol oxidase, PO. Yeah. Oh, this is just a revision, just in case that we don't remember what is phagocytosis. Yesterday, when I prepared the slide, my 14-year-old daughter was behind me, and she asked me, what student are you teaching now? How old are they? I said, I'm teaching university student. They said, we learned this in our in Malaysia, secondary school is Form 2. In Form 2, she said. I said, it's okay. I must have don't remember sometimes, so I put it here so that I'm sure all of you are aware about what is phagocytosis. Yeah, it's a process which certain living cell called phagocyte ingest or engulf other cell or particles. So this is how crustacean defends themselves by having different mechanism. Yeah, one of it is phagocytosis. The cell will so-called eat the pathogen. Yeah, by this process. Slowly they interact with the receptor, and then they release the lysozyme, and then become engulfed inside. Yeah. So this is how they what you call phagocytosis process. Okay, this is prophenol oxidase. It produces melanin and cytotoxic intermediates for bacterial sequestration. So propo is a kind of enzyme in the melanization cascade that also participates in cuticle sclerotization, in wound healing, and also pathogen killing. Sorry, missing the G. Don't worry about this. Yeah, this is just a process, how it is interact with one another, the mechanism of PO activation system in arthropod in crustacean. Okay, another important substance is what we call lectin. Lectin is a glycoprotein that possesses one or more carbohydrate recognition domain, which can reversibly and specifically bind to certain sugar molecule. At the moment, these are the 7 group of lectins identified in shrimp. There are different types: C type, L type, P type, M type, and then they have fibrinogen-like, domain lectin, galactins, and calnexin or calreticulin. These lectins have different structure, diverse expression patterns, and multiple function in the shrimp immune response. So I share this so that when you read journal articles or books, you know, at least you know some background what is lectin suddenly appear. These are the mechanism, the cell, the substance that a crustacean will have to defend from disease. Okay, this is just a table to show you effector defense mechanism in crustacean. So you know already what is propo. There are antimicrobial protein, phagocytosis, encapsulation, lectin, and clotable protein. So what are the cellular population involved? There are semigranulite, for example, hemocyte, when propo involved, and then the target, for example, bacteria and fungi. And then when antimicrobial protein used to defend the pathogen, hemocyte with granules will be involved at cellular level to fight the pathogen. And then phagocytosis, they are hyalinocyte and also semigranulocyte at cellular level. And then encapsulation, semigranulite hemocyte with big refractory granules. So big, when it is big, they use this mechanism. When it's smaller than 10 micron, they use phagocytosis. Lectins in the cellular level, hyalinocyte, semigranulocyte, and hemocyte will be involved. Distinguish between the non-supporting inducing agglutinin and phagocytosis. Okay, so this is the phagocytosis process also. And then clotable protein are from hemocyte to defense from bacteria and fungi. Okay, now we are done with the immune system. A very brief introduction. Actually, immune system is a huge topic. We can have the whole book only on immune system of aquatic animal, but this have been summarized for you just to remember back what you have learned about immune system in fish or in invertebrate, especially in crustaceans. So since the title is about supplement, the effect of supplement in immune system, you have covered, at least you have some understanding on immune system. Now we go to the supplement topic. Yeah. What is a feed supplement? A feed supplement is actually a material used with another ingredient, for example, to improve the nutritive balance or performance of a diet. Feed supplement as the supplement that are added to animal feed in order to fulfill the missing items already present in the feed. So meaning you supply not because they are not in the diet, for example, but they are maybe lacking, for example. The feed additives are the additives that are included in the feed because they don't contain it. You add the additive because the feed doesn't have it. Those additives are added to the feed to boost efficiency or palate it or bring better flavor to it. You know, the word palatable means we make it more sedap. Okay. Immunostimulants are naturally occurring compounds that modulate the immune system by increasing the host resistance to disease. So you will see in the books, in the articles, they use interchangeably feed supplement, feed additive, immunostimulant, though they have slightly different meanings depending on the context of a discussion, but they are more or less with the same purpose. They are added in the diet to increase the performance of your cultured fish, your farm fish. But don't worry too much. So for the purpose of our lecture, I will cover in general the supplement, additives, and also immunostimulant. They are almost the same. Yeah, maybe there are books or article will define it in a different definition, but for this purpose, we just assume that these are supplement that is added into the diet to improve the performance of the farm fish or farm crustacean, your farm species. Okay, the use of feed supplements and additives in aquaculture. Nutritional status is considered one of the most important factors that determine the ability of fish to resist disease, which is true. Have you heard the word, what you eat is what you are? We human, when we eat a lot of KFC, the Kentucky Fried Chicken, for example, Monday, Tuesday, Wednesday, Thursday, Friday, the chances that you will build a lot of fat in your belly, in your body is very high because the fried chicken is very oily, very high fat. So it will significantly influence your nutritional status. So the same thing goes with fish. What they're eating also reflect in their body. So it is quite true with when we said what you eat is what you are. Same thing, not only for human, but also for your aquaculture species. So the increase in cost and short quantity of fish feed have given reason to the need to increase research substitutes, meaning to say, because of expensive fish meal, fish oil, so many research do this kind of study so that we can find a cheaper material, but still perform, still good. So this is the kind of research that the industry, the current researchers are doing in terms of nutrition under nutrition topics. So therefore, the fish feed required to be enriched with additives. Because, for example, you are using too much soybean meal. Soybean meal in the diet, of course, soybean is a source of good protein also, but they are not the same as fish meal. Soybean meal is about 40% protein, but they are plant-based. Do you remember your nutrition class? What is the characteristic of fish compared to the terrestrial animal? Fish has the limited ability to digest carbohydrate. They don't have enough enzyme to digest carbohydrate. So when you use too much soybean meal, there are a lot of consequences, a lot of problem that the fish might encounter later, because they are not meant, the digestive system is not meant to digest plant-based. Fish is supposed to eat. They are carnivorous if marine fish. I mean, most of the carnivorous fish, they are supposed to eat the protein. So when we are trying to develop diet that is too high, for example, with plant-based material like soybean meal, there will be some issues. Yeah. First, of course, they don't really like it because they don't have the fishy smell. And then the nutritional value is not as good as fish meal. So this is where feed additives can help. For example, if we want to use soybean meal, we know soybean meal is lacking in certain nutrient, essential amino acid, where, for example, methionine is lacking in soybean meal. So we can put additives or supplement amino acid in that formula so that there will be not lacking in amino acid anymore. So that is one of the example of the purpose of adding supplement or additives in aquaculture. So feed additives are eatable substances. Of course, they are not toxic. If toxic, why you want to use? Otherwise, you will kill all your fish. Included in fish feed in very little quantity. Normally, when we said supplement, additive, immunostimulant, we don't put like 20-30%. When you put a lot more than 10%, that is no longer supplement. That is part of ingredient. Supplement is added below 10% normally, or even 0 point something if it's very potent, very strong. So it is included in very little quantity to improve the feed, which in return improve the growth performance and decrease mortality. Yeah, even though the topic is supplement on immune system, but when fish grow healthily, immune system will be good also. So there is always link between performance growth and immune response. So when you are able to support good growth with the supplement, normally they will able to defend the disease or increase the mortality of your cultured fish. Okay, so in general, feed additive can be categorized into two classes, which is living and non-living. Let's have a look what does this mean. Okay, the use, okay. The living organism applied as feed are probiotics, plants, and some algae. These are the examples. Yeah? Probiotics are applicable microorganism in feeding the host. Inclusion of probiotic to the feed improve the feed conversion ratio and reduces the death rate. Probiotics are proven to have potential to enhance the immunity of fish, to respond and immune the system to improve the immune system in fish. Of course, in many countries, there are strict laws on the usage of antibiotics and chemotherapeutics in the aquaculture feed industry as a result of bioaccumulation. I'll share you some videos about this. Okay, these are the common supplement additives in aquaculture feed: vitamins, mineral, essential nutrient, probiotic, prebiotics, algae, yeast, enzyme, plants and herb. We will cover on Friday. Yeah, we have medicinal plant and animals. Yeah, the topic on Friday. Of course, bioactive compounds and immunostimulant. When we add supplement, for example, algae, you can use supplement as it is, meaning algae, for example, chlorella, you harvest chlorella and then you dry it and then you make it as a powder. You can supplement your feed with that. Or you can supplement by having to extract the active compound from the algae. This is more potent. This is more strong. Yeah, but of course, it will be more expensive because we have to do extraction. Only certain selected compound that will be extract out and then becoming the supplement. But I am when I do my research, I don't normally do this because I'm not from this background. So what I did in one of my student study is to extract directly and then add in the diet as it is. Yeah. Okay. I want to share. Okay. I want to share about one Biomin video. Hang on, what is the time now? This is quite a long video. Do you want to take rest first, Bu Umi? Question and answer? Or we continue with this long video?
How long it takes, Prof?
Oh, hang on, yeah, I can't remember, but this is a lecture on a feed additive in the diet. Okay, I just want to introduce to you. Biomin is an animal health and nutrition company headquarters in Austria. So they are selling different feed supplement, feed additives for animal, including aquaculture. So back in 2019, they contacted me to run a test for them. Yeah, they are developing additive, enzyme, whatsoever, and then so I agree. We have contract. They pay me under the university, of course, like 80,000 ringgit to run this experiment, and I hire one student to do it, but you know, things not working like we planned because of the pandemic, and Austria and Sabah is so far away. When we send our sample, there are so many hurdles, you know. It end up in Germany. Germany said, oh, I cannot accept, you have to produce certificate, many things. So project is still ongoing with Biomin. Biomin is an international company, very big company, but they have serial lecture on feed additives that I would like to share with the student today, because we have a very long slot of lecture. So I will insert two lecture from Biomin in our slot for this morning. So this is, I think, I don't know, it's almost 1 hour maybe. I can't remember. So yeah, if you have answer a question, I will open it first. Maybe, yeah.
Okay, Professor Sta, I think if it is, I thought it's only like 15 minutes, or if it is 1 hour, probably we will have a pause bit for question and answer, and then you can continue for these videos. Is it all right?
Yeah,
Is it
sure.
fine? Oh, okay. Okay, when there's a questions, please raise your hands, or otherwise I will read it. It's already some questions in chat for the moment. Okay, probably I will read one first and then continue with the raised hand. The first question is from the under S1 students, Dea Andini Rizkyandi. Assalamualaikum, I'm Dea from Aquatic Resources Management Study Program. Permission to ask how to activate the immune system in invertebrates, Professor. Thank you.
I think we have covered some of the what you call in the lecture just now. If you remember the first defense, let's say how to activate, it can be pathogen, or you can create something like toxic condition, or let's say you want to do bacteria challenge test, something like that, or even increasing high temperature also will put stress, and then this will activate the immune system of the animals. So basically, yeah, that is what we can do to activate the natural immune system, because they don't,
Yeah, I think the question will be how to activate. So that it's already answered by Prof Rosita there. Or what factor activated the immune system? I think that's also already mentioned in the lecture, right, Professor?
Yeah, true.
Yeah, about the environmental factor
Yes,
can
true.
activate.
Yes.
Okay, then for second questions, I'll give to Eureka, my Sri Lestari first. This is also from S1. Okay, Eureka, you can deliver your
Iya,
questions.
halo. Sebelumnya apakah suara saya terdengar dengan jelas?
Oh,
Sudah. Oke.
Sebelumnya mohon maaf saya memakai bahasa Indonesia ya Bu ya.
Ya, boleh.
oh boleh. Ya, boleh, bisa.
Boleh.
Iya.
Professor Rosita also understand Indonesians very
boleh
well.
saja.
Oke, Eureka, silakan.
Saya Yurika Maidwi Sri Lestari dari Aquaculture kelas A semester 3 UBPSD KU Kediri. Tadi kan Ibu Umi menjelaskan interaksi antara host dan patogen. Nah, di dalam interaksi tersebut kan ada mikroba yang mempertahankan diri dalam organisme inang tersebut kan ya Bu ya. Meskipun tidak semua mikroorganisme dapat menyebabkan penyakit. Nah, salah satu cara untuk mengatasi hal tersebut kan bisa dengan imunostimulan kan ya Bu.
Ya.
Nah, itu kan bukan hal utama untuk mengatasi atau melawan penyakit tersebut, tetapi hanya membantu mempercepat proses penyembuhannya saja. Nah, karena imunostimulan akan lebih bermanfaat atau berdampak itu pada kondisi di mana sistem kekebalan tubuh ikan mengalami penurunan. Nah, itu bagaimana ya, Bu?
Soalannya, sorry ya, kalau saya bisa tangkap, maksudnya imunostimulan tidak cuma membantu saja begitu, Bu Umi? Macam mana maksudnya?
Ya, how can immunostimulant reacted when the condition of fish is weak?
Susah juga soalan ini ya. I also macam mana saya mau jawab? Sebab kalau yang tadi itu ada disebut pasal microbiota begitu kan? Dia sudah affected by microbe kan? Because kalau ada bakteria misalnya yang immunostimulan kita guna is probiotik lah yang biasanya instead of antibiotics. Because all this while antibiotik mungkin saja sangat berkesan sebenarnya bila kita punya ikan atau udang di affected by misalnya bakteria, antibiotik bisa membunuh infeksi bakteria, cuma yang kita tidak mau dia menjadi resistant untuk masa jangka panjang. Jadi oleh sebab itu kita membunuh bakteria juga dengan bakteria misalnya dengan melalui penggunaan bakteria yang baik yaitu probiotik. Sorry, itu saja yang saya boleh tangkap dari soalannya.
Oke, Eureka, jadi pertanyaanmu kan ketika ikan sudah lemah kita menggunakan imunostimulan, begitu kan? Bagaimana dengan itu gitu? Betul?
Iya benar.
Nah,
tapi
itu tadi
imunostimulan kan lebih berdampak atau bermanfaat pada kondisi ketika sistem kekebalan mengalami penurunan begitu Bu. Terus bagaimana agar ketika tidak harus sesuai dengan kondisi ikan begitu loh Bu.
Ya, susah kita mengatakan sebenarnya misalnya ikan lagi sedang lemah sama ada ikan kalau kita kasih imunostimulan semasa dia sudah infected, meaning dia sudah sakit, sudah lemah, sama ada dia masih bisa atau tidak itu sangat bergantung kepada banyak faktor. Macam juga manusia, kalau kita sudah sakit, sama ada waktu sudah sakit kita baru mau ambil obat, then it really depends on your antibody, your system, whether or not you can defend, right? It's not there will be no straight answer I can and can provide you that definitely they will be able to defend, but it's sangat bagaimana ya, case to case.
Disarankan.
Ya, mungkin tidak sama antara satu ikan dengan ikan yang lain. Ada banyak faktor, mungkin saja environment, lingkungannya, airnya, atau nutrisinya, itu juga menyumbang kepada kekuatan ikan untuk menggunakan immunostimulan yang diberikan untuk menentang the pathogen.
Iya, jadi Yurika, jadi kalau kondisi ikan itu sudah lemah, ya kita harus melihat dulu situasinya gitu. Sebenarnya imunostimulan itu kan memang untuk meningkatkan imun sistem, tapi kalau kondisi ikannya sudah lemah, ya kita harus melihat banyak faktor gitu ya, apa yang menyebabkan, apakah dia bisa menerima imunostimulan itu atau tidak. Terus tadi ada yang dikatakan oleh Prof Rosita bahwa ini kalau misalnya dia terinfeksi oleh bakteri, kita bisa menggunakan bakteri yang baik untuk melawannya juga. Jadi imunostimulannya itu berupa bakteri juga begitu.
Betul.
Seperti itu. Apakah kira-kira sudah menjawab pertanyaanmu?
Iya Bu, sudah. Terima kasih banyak Prof Rosita dan Ibu Umi atas informasi dan jawabannya.
Terima kasih kembali. Oke, next will be Abdurrahman Wahid. Silakan Abdurrahman.
Oke, sebelumnya Assalamualaikum warahmatullahi wabarakatuh, Ibu.
Waalaikumsalam.
Ini saya mohon maaf pakai bahasa Indonesia, soalnya nggak tahu pakai bahasa Inggris. Nah, di sini tadi Ibu sudah menjelaskan tentang suplemen pakan. Nah, di suplemen pakan itu kan, yang maksudnya pertanyaannya saya, jika ikan diberi suplemen pada pakannya, apakah sistem kekebalan tubuhnya itu meningkat? Dan jika suplemen pakannya itu diberi secara terus-menerus, apakah ikan tersebut akan ketergantungan pada suplemen ikan tersebut? Terima kasih, Bu.
Oke, terima kasih.
Ya, silakan, Prof.
Ya, excuse me, sorry. Idenya sepatutnya begitu. Kalau kita kasih immunostimulan, sepatutnya ikan akan berhasil untuk melawan kepada patogen. That's why kebanyakan studi dia melihat berapa percentage kemasukan immunostimulan itu yang akan berhasil untuk ikan gunakan. Jadi bila kamu baca jurnal, artikel atau buku, biasanya penelitian di jurnal, kamu akan lihat, let's say, saya mau gunakan kunyit untuk melawan, supaya ikan saya lebih sehat, supaya lebih rintang melawan penyakit, whatever penyakit yang mungkin biasa menyerang ikan yang kamu ternak, for example. So, if you know the exact study, memang sudah ada penelitian yang mengatakan, oh simpan kunyit 2% of the total diet will give you the very best, then mungkin kamu boleh gunakan 2% terus. Tapi kalau belum ada informasi, misalnya, oh saya akan buat penelitian ini, saya mau lihat berapa persen kunyit akan serbuknya atau ekstraknya akan membantu ikan lebih rintang, or increase the immune system to the itu bergantung tahu dengan apa yang kamu mau lihat, sama ada kamu mau challenge dengan bakteria kah ataupun dengan parasite kah, terpulang apa jenis patogen yang biasa menyerang ikan yang diternak. So dari situ kita boleh tahu. Jadi maksudnya tidak semua immunostimulan itu berhasil bagi semua spesies, bergantung kepada apa spesies, apa yang ingin kita challenge, apa yang ingin kita bunuh dengan immunostimulan itu. Something like that. So soalan yang kedua yang saya dapat tangkap tadi, sama ada, I think this is a very good question. Sama ada nanti kalau terlalu lama makannya itu kita sudah tidak bisa jadi. That one I'm not sure, because this will require a long-term study. Misalnya sudah terbukti itu persen kunyit itu bisa meningkatkan immunostimulan terhadap bakteri. Ya, dia mungkin spesifik terhadap bakteri, and then kita perlu buktikan kalau saya kasih makan kunyit selama 1 tahun, sama ada dia punya performance itu akan turun di tahun-tahun, di hujung-hujung 9 bulan, 10 bulan. I don't know yet. Betul, saya tidak bisa jawabnya itu, tapi berdasarkan logiknya, by right it will maintain the immune system. Macam juga kita manusia, kita kan ambil suplemen apa-apa saja, apa ya, vitamin C. By right, vitamin C akan sentiasa membantu kita. I will not be immune with vitamin C. Logiknya kan, saya tak akan immune. Sudah vitamin C sudah tidak bisa menolong saya selepas saya makan vitamin C 3 tahun. I don't think so in the case of vitamin C, but in the case of other substances, we don't know. So it's very difficult question to answer a general. Tidak bisa kita generalkan, oh immunostimulan itu tidak akan berlaku kalau sudah makan terlalu lama. It depends what is the extract, the bioactive compound in the immunostimulant. So kalau saya kasihkan contohnya tadi vitamin C, rasanya tidak. Malah itu kan essential nutrient throughout the life. So it will not reduce after you prolong the supplement. So itu jawabannya lah.
Ya, apakah Abdurrahman sudah jelas atau ya? Jadi mungkin sedikit aja Bu Umi mengatakan bahwa memang kalau suplemen atau biostimulan itu memang tergantung dari challenge yang dihadapi, ya, penyakit yang dihadapi. Jadi kalau kamu memberikan itu ya sudah dengan riset gitu. Maksudnya di sana patogennya A, makanya kita kasih suplemen tertentu. Itu yang pertama. Yang kedua tadi sudah jelas ya. Jadi kalau kita memberi jangka waktu lama kadang-kadang itu malah sudah tidak imun lagi nanti. Mungkin bisa jadi kebal ya. Kebal. Jadi harus mungkin increase the dose can be also used ya, Profesor Rosita.
Betul,
mungkin.
Betul.
Jadi itu nanti kalau kita memakai terus itu bisa nanti kebal. Kalau kebal bisa harus meningkatkan dosisnya, bisa tergantung itu ya. Seperti itu. Oke, thank you. And the next probably siapa ini? Alicia Syarifah Kusuma. Silahkan Alicia.
Okay, thank you. Am I audible right now?
Yeah, we can hear you.
Okay, thank you. Thank you so much for the change. So I think it's kind of same question like what Abdurrahman already told us, already asked you, but is there any supplements? Is there any criteria on giving the supplements of the immune system on our aquaculture fish? Looking at that, fish have a lot of immune system criteria itself. For example, like in shrimp, we have a different immune system, and in fish, we also have a different immune system. And what time that we usually give the supplements before it already have a disease on that kind of fish itself? Thank you.
Okay.
Yeah.
It's actually, as you know, I haven't go through the most of the slide ya. Nanti ada slide banyak lagi yang akan saya share, termasuk video on probiotics, apa dia punya impact, whatsoever, itu akan didiscuss juga. There are many types of immunosupplement that we can use that may not work in all species. It may work on A, but maybe not on B due to some reason. Yeah, whether immunosupplement from plant-based, like I shared just now, from animal-based also there are some, and then on pure extract, bioactive compounds. So it depends. But if you ask the criteria to me, like I said, aquaculture is a business, so you need something that is profitable. If the supplement is very good but too expensive for you to afford, then you have to think about it. Also, you need to calculate whether or not it's cost efficient or not. So number one criteria is in terms of the cost. Also, you have to consider the cost. Second, of course, the performance of the immunostimulant also. Is it very good? How many species have proven to be, working when they use this supplement? Is there any impact on the environment? For example, using whatever living whatever bacteria or anything, any cell or algae that will have impact on the environment, because we are talking about producing fish using sustainable methods. So you have to consider this criteria when you want to choose what are the supplement. And for us, most likely we have to look for something that is locally available to reduce the carbon footprint. If you have to import from somewhere out overseas, it's not, I think to me, it's not sustainable. It's not, what do you call, it will incur cost and time. Yeah. For example, use what we have locally, our natural herbs, things like that, that the criteria or the basis of choosing a different additive or supplement in your fish farm. So I think that is several criteria that I can think of at the moment. Okay.
Yeah. Okay. Hopefully answer your question, yeah, Lisa.
Yeah, it was. Thank you so much. It's an honor to actually have a great discussion with you. Thank you.
Thank you.
Okay, thank you. Okay, next for the Maudi. Ferrari. Okay.
Hello, ma'am. My name is Maudi Ferrari from the Aquatic Research Management Study Program 2020. Okay, I want to ask about supplement containing immunostimulants can modulate the improvement of the immune system in fish. Well, my question is how the process of this supplement can modulate the improvement of the immune system in fish. Okay, ma'am. Thank you.
Can you repeat your question? Sorry.
Okay.
What is the questions? Maybe you say it too fast.
Yeah.
Maudi.
Okay. Well, my question is how the process of this supplement can modulate the improvement of the immune system in fish.
How?
How?
Okay.
Yeah. How the
yes.
process?
How the process, Professor?
How the supplement
Can improve, modulate or affect.
Okay, if you follow the slide just now, there are several mechanisms for fish or crustacean to defend against the pathogen, right? At cellular level, at first line of mechanism, you remember? So depends in fish or in crustacean, that is how they respond. Their mechanism to respond is the same regardless what are they taking. Meaning to say, if you give immune stimulant, that is to improve their ability to respond. But the way they respond is the standard method through the first line is defend the skin, mucus, or in the gill or thing. And then in fish, if it's a long-term exposure, they will activate the adaptive immune system. What I'm trying to say is mechanism is standard regardless what kind of immune system immunostimulant you use. Immunostimulant will provide the cell, will provide the body of the fish or the shrimp to be able to perform this mechanism. You understand what I'm saying?
Okay, I understand.
Yeah. So that's his answer.
Okay, Maudi, jadi it depends. Jadi tergantung dari di mana itunya ya, diserangnya oleh patogen, apakah di seluler atau di lamellarnya.
Yes.
Ya, dari situlah kemudian timbul proses imun sistemnya tadi, cara melawannya.
Phagocyte lah, apalah encapsulation.
Ya, pertama misalnya mengeluarkan mukosa dulu. Nanti kalau terserang dalam jangka waktu lama, so they will become adaptive.
Very true. Yeah.
Is it clear?
Okay, it is clear. Thanks for the very helpful answer. Thank you.
Thank you, Prof Rosita. And the next one probably siapa nih? Faros. Okay, time is yours, Faros.
Okay, thank you for the chance. So here I will ask about the stress and disease of the fish. Before
Stress
I
and
could
what? Stress and
disease.
feces?
Disease.
Disease. Okay.
Disease, yeah,
Right.
disease. I'm
Yeah,
sorry.
disease. Okay. Stress and disease. What?
And before I go to my question, I will give the background of the problem. So when I went to some village for organization activity, there is the villagers that want to make a aquaculture business, but they have a problem with the thermal situation from that village, that when the night comes, the temperature will be so low, and when the day comes, the temperature will so high, and they don't have any alat, any tools,
Yeah, equipment facilities.
equipment facility to make the temperature become stable. So my question, what can this villager do to make the temperature of the pond become stable without any maybe alat-alat yang khusus gitu maksud saya.
Specific, yeah, specific
Yes.
facilities.
Maybe with the traditional thing, or maybe what? Maybe
Okay, I
Okay.
understand your question. Saya lihat kalau begini kondisinya, masalahnya adalah site selection. When you do your site selection for aquaculture, normally you have to first identify the depth of the site. You know, in this case maybe ini apa ya, sangkar di laut ataupun di sungai.
Di ini aquaculture pond, I think in the pond, Professor.
Kolam. Okay, because
yeah, di kolam.
Saya rasa temperature ini somehow ada dipengaruhi oleh kedalaman air, volume air juga. Kalau misalnya air yang shallow, there is a tendency of fluctuate temperature because the volume of water is not that high. So when matahari panas, senang temperature will be increased. So I don't know any alat except for paddle wheel that can, paddle wheel can mix the water, improve the, what do you call the aeration? Yeah, aeration, oxygen of the water. But normally if it's pond, I would advise the pond should be
covered.
Cover ataupun lebih dalam lebih baik, sebab kalau selalu temperature naik, maksudnya I don't think the pond is deep enough. Ya, itu satu, mungkin tidak terlalu dalam, dan kedua mungkin boleh simpan paddle wheel. Cover, I'm not sure, mungkin juga, but I don't know if if you cover too long, mungkin nanti ada masalah lain yang timbul. That is what I can think at the moment. Or maybe if you put some aquatic plant inside, maybe be able to reduce the temperature also, because they are producing oxygen through photosynthesis, but not to cover the whole area of the pond by having plants lah. So these are just some ideas. I'm not sure. Yeah. Yes.
So Faros, is it clear the professor answer?
Yes, thank you.
Yeah, so you can do several things with the pond. Either make it deeper so that the temperatures would not too fluctuated during the day or in the evening, or otherwise a little bit cover it, or put some paddle wheel atau itu ya apa?
Ya, paddle wheel dan apa?
Paddle wheel itu yang memproduksi oksigen, itu apa sih namanya? Kincir, Masya Allah. Kincir ya, kincir itu dengan adanya kincir itu gerakan air itu juga akan mengurangi suhu pada siang hari, Atau diberi tanaman. You can have an aquatic plant to reduce also heat in the afternoon, or I think that's some ideas for this kind of pond. Okay, thank you, Professor. But maybe this is last to one.
Ada soalan di dalam chat, saya lihat.
Yeah, yeah.
Boleh saya
First,
baca?
yeah,
Ini
yeah.
Vina Febriani. Hello, Vina.
Yeah.
It has physical protection, which can, although it has physical protection, which can get it through the surface of the organ. If there is osmotic stress, can the digestive tract work create a natural immune system? Okay, if you remember the slide, what are the organs involved in immune system? If you see the digestive system, also they have epithelial cells. You know, the cell is coated with mucin, which can, for example, trap bacteria, and then they have also antibacterial peptides, things like that will somehow be able to help, especially when we provide supplement, for example, so that the digestive tract will be able to work better in this case. Yes, of course, digestive system also have some natural immunity also. I hope I can answer your
Yeah.
hang on.
Yeah.
Another
Okay.
question, right?
Yes, another question.
Yeah, Pora Adelia. I'm Adelia from Aquatic Resource Management Study. Permission to ask, can fish be vaccinated? Of course. In Malaysia, there is one tilapia farm, Trapia, which is a very big company. I'm not sure if they are now surviving or not, but it is in Perak State of Malaysia. This is for export. They are used ex-mining pond, bekas lombong, you know lombong Bu Uni, the tin mining punya pond. Yeah, and they use it to culture a very huge like lake, it looks like lake. This tilapia of following high standard for export. So they are vaccinated tilapia. They use this vaccine to vaccinate. Yeah.
Oh, okay.
And fish can be vaccinated, definitely. Okay, and then I saw this Angita Febriani.
Yeah.
Oh, I think I've answered this question in the lecture. Why is it difficult to remove sea leeches from the cultivation system? As we know, this marine leech, they can be a dormant stage, yeah, dia punya so-called eggs whatsoever. Let's say in our hatchery, the system having this small gravel, so they are, you know, hiding in between the gravel. Even though you cleaned it, they are very resistant to if just chlorine, because for example, our hatchery, we don't want to use chemical that much, except when we do total cleanup. So when we clean the coral rubble that become filter everything, there must be some, you know, dormant eggs, what we call, if you can call it eggs, of just that's still alive there. So when we do the culture again with the optimum water, everything is in place, then it bloom again. So very difficult to remove 100% actually. Almost impossible unless we really do a long-term total cleanup, maybe very dry.
Maybe the eggs, Professor, is still there.
Yeah, I think so. Very difficult if it's come to leach, and yeah. Okay.
It's also difficult to remove it from the fish, which is already infected. Oh, it's pity, so pity.
Yeah, geli, sangat geli.
Yeah, really. I don't want
We have a group of researchers in our institute doing this research, so I will share later on their research in the slide. I have the slide for that. I think another question here, how do probiotic work in mixing fish feed? Ini Samsul Muarif, and why can probiotic increase the food conversion ratio and reduce mortality in fish? Okay. As we know, probiotic is the so-called good bacteria, right? So when we have this good bacteria in the gut, and it will have many beneficial things to the host, which is to the fish. So it will improve digestive performance of the fish. When you're, for example, feeding your feed with certain feed, when you have probiotics, I mean, if this work on that fish, so they will help the digestion process, meaning to say, if the digestion is good, digestibility is good, definitely your FCR will be good also. And when fish eating well, digestion is well, all the nutrient will be absorbed to the body, and fish will be very healthy, and mortality definitely will be reduced. So very straightforward link. Relationship. When fish are healthy, chances of mortality will be reduced also. Yeah.
Okay, the last question probably from chat.
Okay, no, Denisa, Nadila, if fish non-specific immune system is physically impaired against infection, will it be inhibit the ability to colonize microorganism in the body or organ? Apa maksudnya ya?
If the fish non-specific immune system is physically impeded against infections, will it be inhibits the ability? Impeded probably disrupt or broke by the infection, but non-specific immune system is innate, isn't it, Prof?
Yeah.
Non-specific immune system is disrupted. So
dia tadinya kan tadi in our lecture, you remember, first defense is innate, misalnya the skin, the gill, they have mucus. But if that infection, whatever lah, virus or whatever pathogen, if the skin or the whatever, the first defense able to fight, then it's good for the fish. But then if it persists, the first defense cannot defend the pathogen, then only it's long enough, for example, you know, the pathogen long enough, and then only the adaptive immunity will be activated. So that's how it works. First, if fish is attacked by stressor or pathogen, the first defense will do their job first. But if the first defense fail because of the long-term exposure, for example, then the adaptive one will take place. That is the basic of system in fish.
But in case of shrimp, they don't have adaptive, so they have to be like what is that? Improve the immune system, isn't it, Professor?
Yeah, so, for shrimp, they only have non-specific.
Yeah.
They don't have adaptive.
So if it is not working, so we have to improve their immune system, I think.
Yes, is it? True, like, you know, improving the diet. That's why most shrimp is cultured in biofloc area, because biofloc is also good. So yeah, things like that. So it's slightly different for the case of shrimp.
Yeah. So Denisa, to add answer for your questions, jadi pada kasus vertebrate dan invertebrate beda, different. Kalau invertebrate itu mereka cenderung lupa. Jadi innate itu tidak bisa mengingat penyakitnya gitu.
That's why kalau shrimp itu tidak divaksin dia. Setakat yang saya tahu lah, sebab dia tidak ada kebolehan untuk mengingat itu.
Ya, jadi oleh karena itu yang non spesifik itu harus selalu
harus kuat.
Harus dirawat terus gitu. Jadi dari awal, karena dia cenderung lupa.
Betul.
tidak punya adaptif. Jadi tidak bisa dia ability to colonize microorganism in the body itu tidak bisa. Jadi tidak bisa. Ya, Prof, I think when the shrimp is attacked by pathogen or by bacteria or microorganism, they cannot remember unless they are improved.
Yeah, that's why we need to put the environment, water quality, temperature, apa itu harus pas, so that itu tidak akan mengganggu dia punya immune system sebab dia kan tidak ada tidak seperti ikan, jadi itu harus bagus in terms of prevention punya cara.
Jadi Dennis harus kalau yang non-specific organism seperti udang harus kondisinya dijaga terus.
Betul.
Jadi tidak bisa kalau tidak dijaga terus karena dia tidak bisa mengingat atau dia tidak punya adaptive immune system.
Iya, betul sekali.
Thank you, Professor, for all the answer of those questions. And okay, I think we can continue for your second lecture, second phase. And after this, for a while, you will be accompanied by Ibu Febriani.
Okay, boleh.
Nanti I will be back later for probably an hour or something like that.
Technologies allows the schools to focus on key systems deliver to learners and teachers.
Hello there, and welcome to our third and final webinar in the Aquaculture Expert Series.
Silakan ya Ibu Febri, saya off dulu ya.
Baik Bu. Assalamualaikum Ibu Febri.
Thank you.
Oke, saya teruskan atau Ibu ada mau You want to say something?
Silahkan Bu, diteruskan saja.
Okay, thank you Febriani. So, this aquaculture expert talk is very good, because this company designed feed additives, which is the topic of our lecture today. So this talk will be engaging an expert, pakar in this field, which is very beneficial for you to watch, whereby they will design the feed using additive and also premix formulation, targeting the aquaculture species. So I hope you will be able to learn something out of this lecture. It is a lecture like watching a conference or just like my lecture also. Okay.
It's brought to you by Biomin. My name is Benedict Standen, Global Product Manager for Aqua, and I'll be the moderator for today's session. I'm joined in this session by Mr. Pete Verstraete, Managing Director of 4C Consulting. He will be talking about feed design through additives and premix formulation. In his role, Pete's providing consultancy services across Middle East, Asia, and also Europe. So thank you very much, Pete, for joining us today. Before we start, I'd like to remind you, the live audience, of two main points. This is an interactive webinar, which means two things. Number one, there will be 3 live poll questions coming through the presentation. This will consist of 1 question and 4 or 5 multiple choice answers. And you will be able to select one which best selects your scenario in your own local region. And this will form the basis of our discussion today. Secondly, you will also have the opportunity to ask Pete as many questions as you like, and we will try and get around to these at the end of the session. So please use the chat function in the GoToWebinar toolbar. Before we start this webinar, I'd actually like to start off with a poll question to get an idea of how dynamic and active our audience may be on this specific topic. So the first poll question will hopefully be shared with you now. And the question is, how often do you develop a new concept for your aqua feed? Is the answer 0 to 3 months, 3 to 6 months, 6 to 12 months, or every more than 12 months? So this is developing a new aqua feed, perhaps if you're working in a feed mill, or if you're working in a field, maybe you change feed suppliers, or you change the way you feed your animals. So we wait just 3 or 4 more seconds for this. Okay, I can close the poll now, and I will share with you the results. So, Pete, for your information, 18% of our audience are changing aquafeed concepts every 0 to 3 months. 14% selected 3 to 6 months. The majority, that's 43%, selected 6 to 12 months, and 25% selected 12 months or greater. So, Pete, perhaps I can ask you what your comments would be on this. And then after you've made your comments, please feel free to start your presentation.
All right, thank you, Ben. First of all, thank you for the introduction. On this poll, I see that it's quite logical. I think once you develop a new concept, you try to stick to it for a couple of months or a couple of, even sometimes a year, year and a half, and try to optimize within the concept. It is very different if you optimize every day for a feed plant. For instance, within the concept, with new raw materials coming, you will optimize your formula for every production just to optimize the cost within the same concept. So I think the result is quite logical to me.
Okay,
thank you, Ben, for your introduction, and welcome to everybody to this webinar. Good morning to the people in Europe. Good afternoon to the people in Asia. It is with great pleasure that I will share some time with you on this topic about aquafeed design. And more in particular, I will go into the role of additives and premixes, the importance of these components in your aquafeed formulation and production. As we go through this presentation, you will see that basically through additives and premixes, you can produce any kind of feed anywhere in the world with limited access to quality raw materials. We will go through the aquafeed design cycle. And of course, the starting point is to clearly define your product and customer objectives. Which species do we want to feed? What is the target price quality? Do we have to take into account any certification or some other constraints like color, smell, anything else from the feed? Once we have defined the objective of the feed, we run through the aquafeed design cycle. First of all, looking at the nutritional requirements. Second, taking into account available raw materials. And 3 and 4, looking at the culture practices and the environmental conditions under which the species is cultured. Once we have done all that, we will enter all the data in our constraints in our formulation matrix, and we will optimize for a least cost formulation to obtain the optimal price and quality of our target feed. Let's go through every step of the aquafeed design cycle, and I will elaborate on every point and give some examples. And we'll start with the nutritional requirements. Defining the nutritional requirements is still one of the major challenges in aquafeed production. Many feed companies struggle with it rightly because we are culturing a vast range of species with very different feeding habits and cultured into very different salinities. We go from very herbivorous fish to absolutely carnivorous fish, and we go from zero salinity to 35 ppt and above, and from ponds to sea cages. Some species like Pinnaeus vaname are even cultured in a wide range of salinities, moving the culture conditions away from their natural optimum, which will of course impact the requirements of the animal. Secondly, in aquaculture, the next challenge is that for every culture stage for different species, we have very different requirements, and we need very different feeds. You can see from the product example of Pinnaeus monodon that in a span of just 4 months, we use very different requirements for the feed, very different particle size. We will need some very specialized raw materials for hatchery feeds or broodstock feeds, and we use different processing techniques. This making our product design not very simple. To understand the nutritional requirements for aquaculture, we basically rely on research done on lab scale and compiled in NRC of 2011. But often this research is done on small animals and only during a couple of weeks. So we may get an indication of the requirements in lab conditions, but it remains a challenge to know what are the requirements for optimal performance under practical conditions and during the complete culture cycle. Even more importantly is to understand what happens with our requirements due to the genetic improvements realized on the species over the last decades. Improvements in growth rates for the main aquatic species have been faster than terrestrial animal selection. As shown in the graph, growth rates for tilapia have risen by 350% since 1985, while salmon rose 270% since 1975. Shrimp 240% since 1990. It is obvious that our aquafeeds need to be adapted to cope with this higher growth potential. I can demonstrate the impact of genetic improvements with the example of a pin monodon farm that switched to domesticate and improves broodstock in the late 90s. Together with the domestication program, a feed program was developed to match the feed specifications with a higher growth potential. And as you can see from the graph, the results were impressive. Average growth rates, including all sizes at the farm, rose from 1.4 gram per week to average of 2 gram per week in a period of just 7 years. This more or less corresponds with the reported growth improvements of 10% per generation of selection. Now, what is the impact on our requirements? Some research is confirming the increase in nutrient requirements with genetic improvements of our animals. For instance, this article from Dr. Lawrence reported a methionine requirement of above 0.8%, much greater than the previously reported 0.6% of methionine requirement for genetically improved P. vannamei. Sorry. The next step in our design cycle is to look at the available raw materials with respect to quality, price, and sustainability. Of course, this brings us to question our dependence on fish meal and fish oil in our formulation of high-performance diets.
Pete, since you're on this topic, you've introduced some considerations which we have to bear in mind when we're choosing raw materials, thinking about formulation, etc. So I'd like to open this opportunity to ask the second poll question to our live audience. So this question should be being shared with you now. And the question is, which factors do you consider most important when choosing a feed formulation, if you're perhaps on the formulation side, or if you're more on the production side, a feed supplier? And the options would be raw material quality, the cost, relationship to the supplier, considering trust, reliability, availability perhaps, or how that raw material or feed will be performing on the field. So please, if you can select one which best suits your own opinion. And again, we wait 4 or 5 seconds. Okay, the majority of people have voted. So I will close this poll now. And Pete, the answers are being shared with our audience now, and it comes as the following. So the majority, the largest share, 48%, chose raw material quality as the most important consideration. 6% are focusing mostly on the price, the cost. 8% think relationship to the supplier is the most important, and 38% are looking for performance on the field. So may you make some comments about these results?
Yeah, it's a surprising result, Ben, because everywhere where we go as a feed supplier, you hear about price, of course. But I think it shows the insight of our audience. I think the most important criteria is, of course, the performance on the farm. But you cannot judge it immediately. You need time to evaluate it. And that's why it's easier to work with integrated companies like often my clients are. We have the time to develop the new concept and verify the performance on FCR, shrimp quality, fish quality, and growth without having the stress of having to sell immediately to third party. I think it's very different for a feed mill selling to farms. There, of course, you have to convince with other criteria that your quality is already good before they have all the feedback on the results. So it's good to hear that raw material quality is considered as one of the key points, because effectively it is one of the crucial points of the feed.
I would completely agree. I think it's very encouraging for us to see that people are willing to invest in a better quality product if they get a better return on investment. Thank you very much, Pete, and please continue with the presentation.
So we were on the point looking at our sustainability of raw materials. And of course, in aquaculture, we are aquafeeds. We are heavily relying on fish meal and fish oil to design especially high performance diets. Now, I will not go into detail in the fish meal and fish oil substitution, as I did that in a presentation for Biomin last year. I will just highlight some points, important points that are often overlooked. So why fish meal and fish oil are so essential to us? If we look at the protein, of course, fish meal is a very rich source of essential amino acids, with the protein levels going up to 72%. But more importantly, the essential amino acid ratios in fish meal are very close to the ideal protein for our fish and shrimp. And the ratios between the various essential amino acids are crucial in our feed design, and fish meal simply represents the best option to obtain these ideal ratios. Furthermore, the soluble protein is giving us higher digestibility, higher attractivity, and palatability in our diets. If we look at the lipid fraction, of course, fish meal and fish oil supply us with essential fatty acids, basically DHA, EPA, who are essential for our marine fish and shrimp. But fish meal is also a very rich source of cholesterol, which is essential for shrimp molting. And it's a good source of phospholipids, which is an emulsifier of underestimated importance in high-performance diet. Looking at the digestible energy, it is rare to find a raw material that is so highly digestible and very rich in both protein and energy. And this is crucial for developing or formulating high energy diets, such as salmon feeds and marine fish feeds. So as we understand the composition of fish meal quite well, and we are able to replace most of these nutrients, the question remains why we are quite successful in replacing or reducing fish meal, even formulating without fish meal and fish oil in lab scale trials, and we still get big challenges when we extrapolate it into the field. Several components present in fish meal may not seem essential for growth, but do affect the performance and the health of the animals. EPA and DHA have an effect on fish health and membrane fluidity. Some nutrients present play a role in liver protection and osmoregulation. Sorry. I have a problem with this. Okay. Phospholipids are crucial in lipid digestion, and solubles and fish meal are very rich in nucleotides and free amino acids, having an impact on palatability and attractivity, and possibly on health. So it is crucial to replace or compensate all the necessary nutrients when we take out the fish meal or reduce the fish meal and the fish oil. But this is not everything. When we replace the fish meal, you have to look also what you bring as an alternative and manage these alternatives and some negative aspects correlated to these alternatives. We have to manage the possible impact of all these other components like plant raw materials and animal raw materials. Many plant raw materials contain anti-nutritional factors and phytic acid. Secondly, many plant protein sources are further removed from the ideal protein and can create nutritional imbalances or amino acid ratios that are not fitting the requirements. Some high fiber raw materials, plant raw materials, and high ash animal raw materials provide a lot of indigestible matter, and this can affect the digestibility and pollute the culture environment, and providing organic matter to potential pathogens to thrive. And last but not least, oxidated oils and fat and mycotoxins affect the immune system and can cause inflammation of the gut. All these negative factors may not kill your animals immediately, but the full cycle effect on performance is not negligible. As an example, the cost for growth due to the production of antibodies was up to 3%, and the cost of growth due to inflammation was reported to be between 10 and 30% in poultry. Now, we don't have this exact calculation of the cost of growth due to inflammation for our species, but we do have quite a lot of data on the direct effect of mycotoxins potentially causing the inflammation on several species. This was nicely summarized. Some of the main results were nicely summarized by Michele Mucchio and Roy Rosen in the Aqua Days in 2019. They reported that DON caused a 10% reduction in final weight and higher FCRs in trout. FUM was having a negative effect on weight gain and an impact on the immune system for carp. And Fusarium mycotoxins reduced the weight gain and increased FCR for red tilapia. FUM was causing atrophic and vacuolization changes in the hepatopancreas of white shrimp, and it was suggested that it had an effect on the muscle structure. So my main key message in fish meal and fish oil replacement is not only to compensate and balance all the nutrients taken out, but also manage the negative impact of the alternatives used. Of course, a major impact on today's feed design is the change in cultural practices, notably the shift to more intensive system and the disease threat that we have from white spot, EHPN, AHP, AHPN, and now DIV1. As a reaction to the disease stress, many farms have a small pharmacy at site to remedy all problems, including bad feed quality. As Dr. Prakam explained in his very nice webinar some weeks ago for Biomin, the key to successful disease management is a holistic approach, taking into account clean seed, clean water, and clean ponds. Only once this approach is in place, the feed can further strengthen the health of the animals for optimal performance. Now, in the new culture systems, it is absolutely crucial with increased biosecurity for the feed not to add additional nutritional stress to the animals. This additional stress can be caused by imbalanced feed and not to pollute the water and the pond due to uneaten or badly digested feed pellets. As a positive contribution to the health, we can fortify the feed with different components according to the circumstances. Immune stimulants such as beta-glucans work well, but need to be applied punctually and in anticipation of a stressor. Stimulating the immune system comes with a nutrient cost, so your feed should be designed taking that into account, the oxidative reactions with immune stimulation, and also the energy required. Because of the risk of overstimulation when you apply continuously, immune stimulants are often limited in practical use to larval feeds and functional feeds. Probiotics have proven to be very effective applications to the pond bottom and water column, and in-feed solutions will continue to gain importance under the current more controlled culture systems such as RAS systems. The reported benefits are important if you can apply the probiotics in the right way. The gut health improvers such as essential oils and organic acids have proven to be very cost-effective solutions, as they can be applied during the whole cycle and prove their benefits even in the absence of a health threat due to improved FCRs and better growth. In all cases, and that is one of the key messages, it is recommendable to integrate all these components into the feed rather than to apply it on the farm. Now, to be able to apply it into the feed correctly, of course, the producers of these additives are making huge effort to stabilize the product better and to resist against the harsh processing conditions that we generally apply into aquaculture feed production. They do that by applying different processing techniques such as encapsulation. But I believe the feed mill has to upgrade also their systems. We have to work with microdosing system to be able to apply or integrate these components in low inclusion correctly in our feed. Our mixes have to be upgraded to mix homogeneously. And one of the important points is that we should, at all costs, try to produce at the minimum temperature possible to still get the stability of the feed or the floating characteristics, but maximally protect all our nutrients, including these functional components. For enzymes and probiotics, we might have to go and look into the vacuum coating system and have a very professional application of these components into our feed. The next step in our feed cycle is to take into account environmental conditions. An important example which I want to mention here comes from shrimp culture in India. Due to the prevalence of white spot, pond water is heavily chlorinated before stocking and is topped up only with borehole water during the cycle. This causes not only low salinity, but also creates mineral imbalances in the water and pH and alkalinity problems. As a result, shrimp are stressed and show signs of cramping. One solution adopted by the farmer is to add massively salts into the water. They hang back into the pond and wait for the salts to dissolve. However, a much more efficient solution we found is to integrate these different salts or minerals lacking into a premix and into the feed. At least we solved the mineral-related cramping problems, but cramping is of course also caused by other problems such as fibriosis. An example from the impact of environmental conditions on our feed design for marine fish is the adaptations to be made when we are confronted with decreasing temperatures. Several stresses occur at the same time and affect performance and can cause serious mortalities. The effect on the animal is threefold. The immune system of the fish is suppressed due to the lower temperatures. The digestive and enzymatic capacity is also reduced, which limits the digestive capacity of the fish. Membrane fluidity is affected, which has an impact on the osmoregulation and causes osmotic stress. At the same time, as the animal is weakened, opportunistic bacteria develop into the system, into the environment due to the lower temperature and can attack the already weakened animals in a violent way. Now, adapted feed solutions addressing each stressor, immunity, digestion, membrane fluidity, and trying to protect the maximum against these opportunistic bacteria have been formulated in the diets and have been tested on marine fish such as gillhead sea bream and on tilapia successfully. While going to our feed design cycle, we realized that the gap between the nutritional value of our raw materials and the nutritional requirements and need for functional components in our feeds, that this gap is widening. Therefore, the use of additives and premix is becoming more and more important in our feed design. So to deal with this gap and trying to fill up the gap, we can formulate different kind of premixes, different functional objectives. Of course, we always use basic premixes containing vitamins, trace minerals, antioxidants, and sometimes anti-mold separate or included into the premix. To deal with the nutrients we take out while reducing fish meal and fish oil, we can formulate premixes containing various minerals, microminerals, amino acids, solubles, taurine, inositol, cholesterol, phospholipids, attractants, whatever you need to deal with all the points mentioned that you have to balance when you reduce the fish meal, fish oil inclusion in your diet. To correct for the quality of the alternatives used when you do the fish meal reduction, you can choose to use enzymes, toxin binders, antioxidants, liver and hepatopancreas protectors. For the environmental adjustments, we can include in our additive packs or in our premixes osmoregulators, selected minerals, anti-stress components, and components that improve the membrane fluidity. Of course, last but not least, is the health additives or the additives or packs that can be used to improve our health situation, together with our holistic approach applied at the farm. We can use simple vitamins and mineral boosts. We can choose to include immunostimulants, probiotics, prebiotics, organic acids, and essential oils.
Pete, may I interrupt you at this point? I think this is a very nice way of categorizing different types of additives, and depending on their positioning, whether it's for nutritional benefits, health benefits, quality control, or even for the environment. So with these 5 categories in mind, I would like to open up the third poll for our audience members. The question would be, what additive groups would you be most willing to explore or apply? Perhaps if you're working in a feed mill, what would be used in your feed? If you're working on the farm, what are you using? Or if you're coming from an R&D background, which would you be most willing to test in your experiments? Would it be the basic premix? Would it be additives or functional ingredients for reduced fish meal, fish oil? Would it be ingredients to ensure better quality or correct quality changes? Ingredients to improve the environment? Or would it be focusing more on health? So we've got a lot of votes coming in now. Again, just wait 4 or 5 more seconds. Okay, we have a clear winner. And then we have a few which are very, very close. So I will share with you the results now. And actually, I think this ties in very nicely with what we were hearing from Dr. Prakan and Dr. Bazzano in the previous presentations. It seems, Pete, that health additives are most interesting for our audience. 46% of them chose this additive category to be the most interesting. After this, we have very close between quality correction at 16%, reduced fish meal, fish oil at 17%, and basic premix at 14%. And just 7% of our audience are looking at the environmental adjustments. So as I say, a clear winner with the health additives, followed by, I would say, additives with a more nutritional positioning. What do you think of these results, Pete? Is this what you would expect?
I think it reflects the status of our industry and the worries of our farmers and producers, which is, of course, very much determined by the health. It also, I think, indicates that some of the additives have proven or are proving their value, and people have made a shift in their minds where, let's say, 15, 20 years ago, we would not think so much of health additives. Now this is becoming more important. I think also it has to do with our progress we made on the, let's say, normal nutritional factors. We are maybe less worried about how to reduce the fish meal and the fish oil a little bit, and we are much more worried about, let's say, the health and how it works together with a good diet.
Okay, thank you. I agree. I think functional feeds have a very important role to play in the future.
Yeah. The environmental adjustments are maybe more difficult to manage because many people need to apply just punctually. So it's finding in a more difficult way, finding its way to the farmer.
Okay, perfectly agree. Thank you, Pete.
Okay. So after defining the several options and premixes and additive packs that we can design or that we can choose of to close the gap between our raw materials available and our objective of our feed, I will go into some practical examples just to show, okay, how we should reason, how we put into work our feed design cycle, and just some different examples from very different applications. The first practical example is from rohu carp. The objective of the feed, which is the first thing we should look at, of course, the objective of the feed and the customer in this case is a very classic one. I get this question very often, and I'm sure many of you get this question too. Can we make a feed that is cheaper and better? Now, if you look at the original formula applied, then you can see, first of all, if you look at that formula, we analyzed all the raw materials to make sure we have the right composition and to detect eventual problems with some of the ingredients. We analyzed all ingredients, plant ingredients on mycotoxins, for instance, and we found a very record high level of aflatoxin in the peanut cake, up to 9,000 ppb. Secondly, if you look at that formula, the customer is relying very heavily on rice bran, which is a raw material which is cost effective for sure, but often has quality risk, especially with regarding to the fat quality and oxidation related to that. So thirdly, this formula had not been optimized. That's where the poll in the beginning is important. This formula had not been optimized for many years, and often some feed plants active in especially freshwater fish do not optimize their formula regularly. So you can be pretty sure that with the actual raw material prices and qualities, you will get a better optimization when you redo the exercise. So what did we do? Following our cycle, we said, okay, of course, first of all, you have to look at the nutritional requirements and rebalance most of the nutrients as good as possible. As an example, first thing to look at is also the amino acids and the amino acids requirements and the ratios between the different amino acids. So we compensated and increased the limiting amino acids, methionine and lysine, in first instance. Secondly, you have to look at your raw materials and what is the risk they are bringing. So we reduced the inclusion of rice bran and reduced the inclusion of peanut cake to limit this risk or quality risk on the raw material. After there was still considerable mycotoxin risk because all of the plant raw materials had some levels of one or the other mycotoxin. So we included, to correct the quality of the raw materials, we included the mycotoxin binder and the feed preservation product. As a result, and the optimization done, the cost was reduced by 9%. And overall, the nutrient levels were higher than before. As an example, you can see the lysine and methionine levels that have increased compared to the first solution. So our objective of cheaper and better had been obtained by simply going through the design cycle.
Prof Rosita. You didn't hear the sound?
Yeah, you didn't hear the sound?
Yeah.
Oh, okay.
Going to decrease the fish meal levels and fish oil levels, but still get this nice performance that we had. 25%. And fish oil from 6% to 3%. Of course, the thing to do was to rebalance first the limiting essential amino acids. In this case, methionine was increased, lysine was increased, and we added threonine to the diet. Looking at the other nutrients, we formulated on the same levels of soluble protein. We increased the DHA level with a sustainable source of DHA, and we increased the phospholipid level on the same level as before. To manage the potential negative impact of our fish meal reduction, we added some additives, like a toxin binder, of course. Here we see. We added a toxin binder. We added betaine, taurine, and for gut health improvement, we added organic acids and essential oils. So the objective, equal performance and survival with less fish meal and fish oil was definitely achieved in that trial. As a last example, practical example, one from pea monodon. Again, a very different objective, not cheaper and better, but to reduce the fish in, fish out ratio, which is a sustainability target. But at the same time, in this farm and this system, you don't want to lose the high performance, and especially don't want to change the same taste and texture. So you cannot do things very abruptly. You have to go step by step. So what we did is to reduce the fish meal from 32.2% to 22.5%. Fish oil was reduced from 3% to 1%. and krill meal from 2.5 to 1.5. You still have to get the good performance because in your fish in, fish out, your CR is of crucial importance. You cannot substitute the fish meal and increase at the same time your FCR, your result is becoming worse. So you have to use a lot of nutrients and other raw materials to compensate for what you take out. The fish meal was replaced, fish meal and the fish oil was replaced by more sustainable sources like fish byproduct meal, squid byproduct meals, and vegetable protein and vegetable oil. Hydrolyzed fish byproducts were also used to compensate for the loss of soluble protein. Amino acids were balanced by increasing lysine and methionine inclusion into the diet. To balance the lipid fraction, it had to be in a sustainable way, so we used the alga prime DHA powder from a sustainable source. Soy lecithin was also increased to have the right phospholipid levels, and we had to add technical cholesterol to compensate for what we take out. Final quality corrections were done by adding a mycotoxin binder, natural emulsifier, and a digestibility enhancer. So as a first step in our fish in, fish out reduction, with the objective of high performance and same taste texture, we achieved this objective, but of course by including a lot of additives and premixes. In the next step, we try to optimize this concept further and reduce the cost. So basically, we can find very practical solutions to optimize our aquafeeds toward objectives by complementing our raw materials with additives and combine the different additives into premixes. As a next point in this presentation, I would like to have a look at the advantages of using premixes rather than separate additives, which is not always easy to apply for our, let's say, basic feed mills who don't have all the capacity for accurate mixing or microdosing. So the first objective, I think you got from the presentation, the first objective for us to use premixes is to offer flexibility to the feed plant to produce and design different feeds with a limited set of raw materials. You can basically design every kind of feed by simply adding a premix into your feed. By using premixes also, you can be sure that all the components, often with very small inclusion rates, can be mixed and dosed very accurately and mixed homogeneously into your feed. Now, what about the price? Using a premix today should be cheaper than buying all components separately because of the purchase power of the premixes. The premix producer is buying his components for several feed factories and has his weight on his purchases or purchasing power. But to me, the main advantage of using premixes today is that the premix companies should help you to overcome some of the difficulties in aquafeed design, as mentioned earlier on, such as the definition of optimal nutritional requirements. Today, premix suppliers can become real partners in nutrition and development to the farmer and the feed mill. As most premix companies have their own R&D facilities to run, of course, their product trials and verify their product concepts, but also they are used to verify feeds or feed concepts for their customers. Also, the premix companies and additive companies are giving formulation support to their customers, optimizing their feed, giving recommendations to the nutrition, and helping together to get the best price quality out of their raw materials. Lastly, the premix supplier or additive supplier is following up on the results with the farmers. He has big connections with the farmers and can follow your feeds and your feed results, including their additives and premixes with the farm where it's tested. To me, this represents a huge added value to the customer, as a premix company has such a vast network and can transfer field experience gathered from anywhere into the world and improve our performance of the feed. So it's time to wrap up. Concluding this presentation, you can say, okay, looking at our available raw materials, we realize that the nutrients provided are becoming insufficient to deal with the increasing requirements for optimal performance of our fish and shrimp. Therefore, we increasingly rely on additives to complement our raw materials in search of fish meal and fish oil reduction, to adapt for environmental and culture conditions, and to support the health of our animals under suboptimal conditions. The use of premixes offers a practical solution to offer flexibility into your feed design. Whereas in the past, we were looking at premixes as just a complement of vitamin and minerals to our raw materials, in the future, we will consider the premix and additives as the key part of our formulation, bringing all the essential functionalities and nutrients to our feeds. And for the companies supplying these premixes and additives, they have a key role to play as partners in nutrition and health, helping us to make better feeds for better performance. Thank you very much for your attention. This concludes my presentation. I believe we have a lot of time for questions. Up to you, Ben.
Okay. Okay, I hope everyone is listening. How was the talk? I find it very, what do you call, informative, because it is from a very trusted, what do you call, trusted website, trusted source. Some of the question asked by the student just now, I think they give the answer here. When the student just now asked, one of the question was, what is the criteria in, you know, taking immunostimulant or something like that? And in this lecture, in this talk, the presenter also lists out different criteria for ingredient. If you remember, yeah, the early slide, when he offered, there was a quick poll survey, right? The speaker opened survey, the moderator opened the survey for the audience to answer. One of the questions just now, if you remember, what are the criteria for the ingredients, yeah, when they choose? So the audience in this talk is mostly those are practicing, the farmers, the business person. So when they answer, it is interesting to see that they answer at cost, which my answer to you just now, cost is number one, right? I said, when you choose immunostimulant, one of the criteria is, of course, the cost, you know, whether it's too expensive. I said, you might be, you know, will not getting any profit or something like that. But interestingly, in this poll, in this survey, the audience choose cost as the last one. The first they go is the quality, if you remember. So the opposite what I share with you, and then when the speaker will ask what is his opinion, he said he also surprised because he also expecting cost as one as the top criteria in choosing immunostimulants, which is in this case the audience put cost as the last importance. They put quality is number one. So according to the speaker, they have to be very careful when considering this criteria. So that is one of the things that I'm not sure if you follow that one, but I hope you do. And then there is another poll, another survey, whereby they ask about the different types of additives. You remember the photo? There are 5, right? Different types of, they put the premixes lah, additives or supplement. This speaker categorized basic premix or reduced fish oil, fish meal, quality correction, you know, when you use soybean, for example, or alternative ingredient, and then environmental adjustment. How do you try to save the environment when you feed your fish? And then the last one, the health additives. And very interesting, the audience choose, most important to them is the health additives or the immunostimulant. That is the one who the first criteria or the first types of premix or types of supplement that they will choose. Yeah, so it is very interesting to follow this talk. If you have any question, I might open question for this lecture from Biomin. Do you have any question that you want to ask that you are not clear or you don't really understand? But I think it's a very straightforward lecture whereby they are trying to portray the importance of supplement in fish feed. If now we call it as supplement, supplement means maybe you don't put also okay, if you put even better. But the way he explained just now, maybe in the future supplement is something that is compulsory to be added in the formula. So it is very interesting to me. I hope it's also interesting to you. Do you have any question on this one? No?
Okay.
Yeah, Abdul Rahman.
Okay, please Abdul.
Oke, Bu, terima kasih. Sebelumnya perkenalkan, nama saya Abdurrahman Wahid dari Universitas Brawijaya, Prodi Aquaculture kelas B. Nah, di sini yang saya ingin tanyakan dari video yang saya amati barusan itu tentang mikotoksin. Nah, di dalam mikotoksin itu kan tadi ada maksudnya ada 4 jenis ikan, tapi yang saya ingin tanyakan itu dari 2 jenis ikan. Yang pertama itu yaitu ikan mas dan ikan tilapia merah. Nah, yang pertanyaannya itu mengapa pada ikan mas efek negatifnya itu pada penambahan berat badan, bukan pada penurunan, sedangkan pada tilapia merah mengalami penurunan, dan mengapa keduanya itu berbanding terbalik? Terima kasih, Bu.
Okay, terima kasih Abdul Rahman. I'm afraid that I will not be able to answer your question because I don't have the data or a full paper from this study ya, yang ikan mas tadi kenapa efeknya berat dan tilapia pula penurunan. I'm sorry I don't have access to that publication. I believe apa yang dia share tadi adalah berdasarkan studi ya, is based on a penelitian yang dibuat oleh researcher lah, whoever researcher there is. So that is the result from, there must be some factors because the same supplement or immunostimulant might be work in species A but not necessarily work on B or C. That is for sure because different fish has different ability. Maybe they are cultured at different culture condition. For example, ikan tilapia di culture di mana, di tangki, ikan mas di kolam ataupun di aquarium or something like that. So it's very difficult to tell if you don't know the condition of the experiment. And I would say kesan suplemen, kesan immunostimulan itu banyak dipengaruhi oleh jenis spesies yang kita teliti buat penelitian. Stage-nya, we don't know tilapia itu stage-nya tadi itu apa yang besar, apa ataupun larvae kecil, juvenile, we don't know. And then makanan mungkin is a standard factor here, right? Using the mycotoxin punya nih. But then we don't know the other factors like quality of water, even the quality of the fish itself. So ada banyak faktor yang mempengaruhi perbedaan hasil penelitian walaupun yang diteliti itu benda yang sama. Misalnya tadi mycotoxin, I don't remember, fumonisin kah apa yang dia test, might be resulting different result in a different species. Or even the same species, if you repeat, you might not be able to get the same findings. So in my case, yang saya kerjakan dengan biomin itu adalah fumonisin, sejenis mycotoxin, whereby kita mau lihat kalau saya tambah enzim di dalam diet yang ada fumonisin, apakah nanti ikan saya kerjakan lates Riva ASEAN Sea Bass. Apakah ASEAN Sea Bass itu jadinya lebih besar ataupun healthy atau tidak berbanding yang tidak ada enzim, for example. So saya itu develop eksperimen yang ada mycotoxin dalam bentuk fumonisin dan juga ada enzim yang dia ciptakan yang dia tidak kasih tahu saya apa jenisnya sebab mereka ini bisnis, jadi dia jadikan itu satu secret, trade secretnya. Dia cuma mau tahu apa resultnya terhadap ikan itu. Jadi kalau saya bandingkan misalnya yang tanpa fumonisin, tanpa enzim, yang kedua dengan fumonisin dan enzim, yang ketiga dengan fumonisin sahaja, for example. So these three I will compare how is it performing dari segi growth, survival, immune response, maybe in terms of whatever immune response yang saya boleh parameter that I can detect in my lab. So there are many factors that influencing the result, yeah. Even though you are testing the same materials. I hope I answer you, Abdurrahman. Ada lagi soalan?
Sudah Ibu, terima kasih.
Sama-sama.
Thank you, Abdur. Okay, another question from the lecturer, maybe. Okay. I think the video is very clear, Prof.
Yeah, sangat clear kan? That's why I like to share this video also with my students because it is a very it's a based on research. So it's a very good sharing and yeah, I think very useful video and easy for them to understand with the photos, with the written subtitle ada, even though that the speaker is speaking in English, but the subtitle is still there for us to follow easily. So that is a very good I think.
Yeah, right. Make easier to us to understand with the subtitles. Yeah, maybe if there is no question yet, maybe we can take a break, Prof, for the participant maybe wants to go to restroom or some drink.
Boleh.
For 5 minutes it's enough or 10 minutes, Prof?
Maybe
10 minutes, Mbak Febri?
Yeah, 11:30 maybe 11:30 kan? Now 11. Oh sorry, your is 10:30.
Okay, 11:30.
Yeah, boleh.
10:30 we start again.
Yeah,
Okay.
Alright. See you soon.
See you. Thank you, Prof.
Right.
Baik, silakan adik-adik kalau mau istirahat terlebih dahulu. Kita balik lagi 10:30.
Makasih, Dik Febri ya.
Ya, adik-adik tolong jangan disia-siakan ya kesempatan ini, silakan bertanya. Jangan malu-malu.
Dik Febri? Halo.
Ya, Bun.
Ini Zoom-nya tetap nyala yo. Soalnya terrecord per jamnya. Oke. Thank you.
Tetap dinyalakan, Bu.
Oke, matur nuwun ya.
Nggih, Bu.
Dek Febri, hari
Iya.
Jumat tanggal 8 ya. Nanti kalau Ibu ke LP3 jenengan siap ya, sama Dek Dai ya. Bisa?
Bu.
Bisakah? Bisa.
Iya, bisa. Ntar saya ini aja, Bu, kelas saya online aja, asinkron aja. Ada kelas kebetulan hari Jumat. Nggak apa-apa.
Kelasnya bawa ke sini aja, Bu.
Bukan, aku kan bukan kuliah. Aku ke LP3. Tanggal 6 sampai site visitnya Akasa. Halo.
Oh iya.
Ibu ke LP3. Kalau ngajar iso asinkron, LP3 ngurusi itu loh, Akas.
Iya Bu, maksudnya kelasnya Bu Febri itu, Bu.
Kelas saya maksudnya. Ntar saya kelasnya
Maaf. Iya, aku
Iya, nggak apa-apa.
Anu wes, Dek Dai sama Dek Febri ya, siap ya. Nanti bisa berjoin ya.
Gampang Bu itu mah.
Ya, wes. Ini kita kembali jam berapa? 10.30 kah?
Ya, 5 menit lagi.
Oh, iya.
Comeback Bu Umi.
Ya.
Selamat datang Pak Mul.
Ya.
Jangan lupa nanti jam 1 ya di MSP.
Loh, Bun.
Dalam?
Yang ngadakan MSP sama pasca ini MSP juga yang ngadakan.
Panitia saya itu ditunjuk judulnya itu.
Berarti wong MSP
yang
apa ya
MSP
yang ngadakan
itu ketuanya Bu Septi yang MSP. Iya. Tapi yo intinya yo anggotanya dari S2 BP yang
Oh, gitu. Ya.
Karena double nyambi itu loh IS Kak.
Kok anu ya? Kok tumben kok nggak ada dosennya kita gitu? Biasanya
Kemarin
ada
Pak
loh ya.
Kemarin Prof Maftu minta cepat karena terkait dengan SPJ nanti tidak nutut.
Loh, ini dengan yang lalu sudah 1 tahun ya Bu Umi?
Sampun. Yang
Cepet
lalu
ya?
kan lah ya itu kayak 3 minggu lalu toh.
Iya ya, cepet ya.
Nggih, ya cepet toh.
Bisa tahun ya.
Iya. Yang tahun lalu kita kan MSP kebagian 2 toh. Kebagian 2.
Ya, tahun lalu pun juga nggak ada dosen kita, Bapak Ibu. Jadi memang fokus ke visiting professor sama yang dari praktisi.
Oh gitu ya. Kemarin itu ada komen tuh di Guyub Rukun dari Prof Sukoso, ini acara apa ngono kok luar negeri kabeh.
Ya itu positif. Mbak ya tanya dulu ya.
Nek 3N1 kurang 1 memang. Ini 2N1 berarti.
Sama moderatornya Pak satunya.
Bannya kita. Iya, kan sudah Bu Umi loh. Pas
enggak, tapi anu loh, banyak dari kita itu sebenarnya yang sudah capable untuk jadi narasumber gitu loh, maksudnya itu. Mungkin
Cuma
Prof Diana
terus
atau siapa.
Yang dari kita kan di kuliah rutinnya, Bu.
Kenapa?
Yang dari kita kan di kuliah rutinnya, misalnya mata kuliah imunologi molekuler. Dari 16 kali pertemuan itu kan beberapanya masuk di 3-in-1 gitu, Bu.
Iya, mungkin mau yang dari kita mungkin formatnya webinar seperti yang lalu itu bisa.
Betul, iya. Itu kan lumayan menambah itu ya. Kalau misalnya beliau-beliaunya itu menyampaikannya di kelas biasa, terus ini dijadikan webinar walaupun sama platformnya Zoom atau Meet ya lain itunya kesannya. Jadinya
Bu Umi, cuman Bu Umi tidak bisa di SPJ-kan lah masalah tuh.
Oh gitu ya.
Nah ini kan masalah.
Itu.
Tidak
aja
bisa.
Bun. Maksudnya itu memang tidak bisa di SPJ-kan, tapi nggak ditawari, mau nggak usah pakai SPJ, tapi kan dapat apa namanya sertifikat sama itu SK. SK sertifikat itu lumayan loh itu.
Ya, tapi harus membuat panitia dulu Bu Umi. Lah ini kalau dari rektorat ya pagunya SOP-nya begitu.
Oh gitu ya. Jadi memang dari visiting professor
dari pusat, iya.
Iya, kemarin itu maksudnya 3-in-1 itu kan ada visiting professor, terus apalagi Bu, tiga dalam satu tuh 3-in-1 tuh lupa saya. Kegiatannya
publication nggak sih? Join publikasi. Ya belum ya?
Enggak, kalau enggak dulu kalau nggak salah itu ada satu lagi itu kayak writing scientific writing ya Bu Un ya dulu deh.
Ya. Oh yang semacam klinik gitu ya Bu?
Iya, tapi dengan catatan langsung itu gitu, langsung publish. Kayak gitu. Tapi nggak tahu ya, mungkin bentuknya agak beda sekarang ini, yang sekarang ini. Bu.
Sebenarnya itu Bu, itu tergantung konsepnya. Kalau tahun lalu memang ada diskusi manajemen juga, ada diskusi publikasi. Dan kalau outputnya bisa milih antara publikasi atau kerjasama institusi. Jadi tergantung kepanitiaannya sih sebetulnya.
Gitu ya. Eman saja nih kalau ada iya ya. Saya tuh sering sudah ngikuti scientific writing-nya langsung dari Elsevier itu ada. Jadi sekali ikut itu terus ditawari terus untuk ikut gitu. Saya sudah kalau nggak salah udah 3 atau 4 kali ikut langsung dari Elsevier sendiri, dari editornya, dari jurnalnya, ada dari itunya reviewernya gitu.
Ya itu menarik Bu kalau itu.
Sebenarnya bisa
itu yang kalau saya sendiri sih memang yang tahun lalu itu ternyata semua prodi membuat scientific writing itu juga waduh sama aja nih.
Lah ya itu kalau satu sudah itu ya udah satu aja jadi bisa ikut semua di situ gitu. Semuanya sama gitu. Jadinya itu. Oke. Oh, hello again Prof Rosita.
Oh, kembali lagi.
Kembali lagi. Still about 1 hour more, little bit.
So, anak-anaknya sudah semua kembali, belum?
Iya.
Prof Rosita.
Waalaikumsalam.
Oh, sudah pada balik.
Sudah?
Iya.
Bisa kita mulai sudah? Atau
silakan Prof.
Okay. So, we continue ya. We continue the video. Now we are looking at the individual supplement. Yeah, it's by case study based on what people publish so far. Yeah. Okay, the first case study is on the effect of vitamin C on immune response of fish. I myself in my lab doing this with my PhD students on hybrid grouper. So basically, vitamin C is one of the vitamins that is very, very important to boost immune system in fish. So that is why I think if you check on vitamins on immune system, vitamin C appear more frequently than any other types of vitamins. Yeah. So basically, vitamin C biosynthesis does not occur in fish due to the lack of the last enzyme of the biosynthetic pathway, which is the L-gulonolactone oxidase. So therefore, we need to put or to include vitamin C in the diet. Vitamin C is one of the strongest antioxidant defense system in fish. It acts against intracellular and also extracellular reactive oxygen species. And vitamin C does not act alone. It collaborates with other antioxidants, for example, vitamin E or any antioxidant enzyme to further strengthen the body antioxidant defense system. So there may be a case that vitamin C interact with vitamin E. So in my lab, I studied vitamin C alone and then vitamin E alone, and then I also studied the interaction between vitamin C and vitamin E effect on immune response of the fish. Okay, let's have a look at this paper. This paper is investigating the effect of dietary vitamin C on immune response of Atlantic salmon. Yeah, don't worry, although we don't have salmon in our country, but the way they run the experiment, the concept, the mechanism is still the same like in other fish, including our tropical fish. So in this study, they are using the juvenile salmon par provided with diets depleted on or supplemented with normal and high. So meaning they were testing 3 levels. One is considering 0, but other ingredients like fish meal or whatever ingredient used in this study may contain some vitamin C. So even though we don't put any vitamins, when we analyze the diet, we may end up with some amount of vitamins in the diet when we do vitamin C analysis. So actually what they do is they don't supplement the diet with any vitamin C, which is the control, 0.05 gram, and then they have one supplemented with, they call it normal, about 0.31 gram per kilogram of vitamin C, and the other one is on the higher level, which is 2.75 gram per kilogram vitamin C. So after 26 weeks of experiment, they take out the liver for analysis of vitamin C content. And then they also do serum protein level and differential leukocyte counts appear to be unaffected by vitamin C nutrition. So meaning they do a blood test lah. Okay. Of the non-specific cellular defenses examined, respiratory burst, which is superoxide anion activity, and erythrophagocytosis were unaffected by dietary vitamin C level. Serum complement activity, however, was significantly reduced in the depleted fish, and high-dose fish exhibited enhanced complement activity compared with levels in fish-fed normal diets. And then specific humoral responses after the immunization with either Aeromonas salmonicida or human gamma globulin and lymphokine production function independently of dietary vitamin C. And on buff challenge with a virulent strain of Aeromonas salmonicida, vitamin C depleted fish exhibit a significantly increased mortality rate when compared to the other groups. So if you need to develop a study, so you can so-called have a look at this experimental design and what are the parameters that they are detecting or analyzing as a result of effect of vitamin C on the immune response. So basically, in all the parameters that they analyze, for example, this respiratory burst, erythrophagocytosis, serum activity, and then specific humoral responses, and then of course bacterial challenge. Yeah, these are the parameters that they studied, that they analyzed, but they are not all affected by vitamin C, meaning putting vitamin C in the diet doesn't have any significant in all the parameters, but some like bacterial challenge, yeah, it turned out that if you put vitamin C in the diet of Atlantic salmon, there will be significant improvement if you have vitamin C, meaning better than if you don't have vitamin C, if the fish affected or infected by this particular bacteria. And then for humoral response, no significant difference. Yeah, but for serum complement activity, yes, there was significantly influenced by the vitamin C. And then on non-specific cellular defense mechanism, not affected by vitamin C. Yeah, Same thing with serum protein, also not affected. So meaning to say, in this experiment, it helped the fish to combat or to defense against this bacteria infection of Aeromonas salmonicida. So these are example of real experiment people conducted to increase or improve immune system of their cultured animal. In this case, using vitamin C on salmon. Okay, this is the second experiment. They are using vitamin C also on flounder. Yeah, they want to see whether vitamin C, you must remember vitamin C exists in different forms. Yeah, some are derivatives. Different form of vitamin C might have different performance. I have tried two different form of vitamin C. One ascorbic acid, the other one is something monophosphate, I forgot. So they are performing differently. Yeah, so in this case, they were working on starry flounder of this weight. This is quite big fish, right? About 139 grams is quite big. And then the diet were designed to have this supply of L-ascorbyl 2 monophosphate. And then they were investigating the effect on body weight, FER, SGR, compared to the control is 0. Okay, in this experiment, I'm not sure, I didn't have the full paper now. This one is not the analyzed level, yeah. This is the, what do you call, calculated level. If they analyze the vitamin C, it may not exactly 50 milligram per gram. It might be different, like the one, the previous experiment on salmon. So the result show that body weight, FER, SGR was significantly higher than the control. Control here is the 0. Vitamin C, meaning if you put vitamin C, your fish will grow bigger, better feed efficiency, and also specific growth rate. In terms of blood cell, the hematology, AA diets increase red blood cell count, hematocrit, hemoglobin, plasma calcium, magnesium, whereas total protein, glutamic, oxaloacetic transminase, and glutamic pyruvate transminase was significantly decreased. So meaning to say addition of vitamin C significantly influenced the hematology of this flounder. And then SOD, CAT, and then glutathione and glutathione as transferase activity was significantly decreased compared to the control group. So last one, lysozyme and phagocytosis was significantly enhanced by increasing dose of ascorbic acid. This result indicated that ascorbic acid diet has a positive effect on growth performance, hematological parameter, antioxidant, and non-specific immunity in starry flounder. So if you want to do, let's say, in your place, you have new species that no one really studied it yet, so then you can develop experiment to determine the effect of vitamin C or any other vitamins on a specific immune system of that particular species. Okay, another one is on the effect of vitamin C on, this is freshwater fish, mussel. I think Indonesia, you have tor, right? Species. We do have our tor, but not tor putitora. We have tor duaronensis in Borneo, and then tor Tambroides in Peninsular Malaysia. So in this study, they have 4 treatments. 0 is the control, 100, 200, 300 milligram per kilogram vitamin C. And then the fish is juvenile, only about 2 grams distributed in a 12 tank. So meaning they have 1, 2, 3, 4, 4 times 3, or they only have 4 treatments. So after 70 days, they were subjected to a stress, not a disease, hypoxia. Yeah. And then fish fed 300 milligram per kilogram vitamin C shows significantly increased serum lysozyme activity in response to hypoxia stress, followed by 200 and 100 milligram per kilogram. Similar results were observed for glutathione peroxidase, glutathione reductase, and superoxide dismutase. So after hypoxia, malondialdehyde MDA concentration was considerably reduced in fish fat 300 milligram per kilogram. So in this study, it concluded that stressful conditions such as hypoxia, when feeding diet supplemented with 300 milligram per kilogram would be better than 200 and 100 milligram per kilogram, meaning the fish obtain the immune and optimum immune system and also antioxidant status. This is quite true. For example, when your fish is under stress, like water quality is not good, there's pollution, there is red tide, the harmful algae bloom happening in the cages, something like that, you might want to introduce diet with extra vitamin C, because it is proven. There's many studies showing that vitamin C serves as a very strong antioxidant in the system and also increase the immunity. That's why even in human also during this pandemic, we were advised to take extra vitamin C because the concept is the same. Okay. Okay, this is our own paper. This is my PhD student. We've done on L-ascorbyl 2 polyphosphate. The first paper was on ascorbic acid. We did it on hybrid grouper, whereby we fed our hybrid grouper with 8 experimental diet. How do I... Okay, containing vitamin C level 0, 18, 45, 78, 40, 144, 245, 386, and 762. So we have triplicates. Our fish is only about 10 grams. It's a juvenile hybrid grouper. And then the final body weight, the weight gain, specific growth rates, and fish survival increase when fed with vitamin C based diet, regardless the supplementation. Meaning as long as starting from 18, fish perform better than those without the supplementation of vitamin C. Total feed intake reduction was observed in fish fed non-vitamin C diet, meaning to say they don't have much appetite compared to those with vitamin C. Whereas no significant differences were detected in the feed conversion ratio and protein efficiency ratios among treatment. Hepatic VC concentration, the liver vitamin C, was positively correlated with dietary vitamin C supplementation and reached a plateau when supplemented with more than 386 milligrams per diet. You know that liver is the organ to store all the nutrient, including this one vitamin. So we analyzed the liver and then we found that the level is positively correlated, meaning the higher the level of vitamin C in the diet, the higher the level of vitamin C in the level, but they are plateau at this 386 milligram. Higher total protein and alkaline phosphate levels in blood serum skeletal deformities were recorded in the fish fed with a controlled diet compared, spelling, to other treatment. So in this study, we do some X-ray to the fish. We found that the bone having abnormality, there are lordosis, kyphosis, so scoliosis, all different types of bone abnormality were observed in fish fed without the vitamin C, but it did not really show much in terms of the weight lah. Okay, the result indicate that adding 18 milligram per kilogram of vitamin C is optimum, and further increase does not provide any significant benefit to the fish and growth. Meaning to say, if I put 386, fish still grow the same as 18. So we will take 18 as our minimum or optimum level for vitamin C supplement if it's a normal condition, but we may want to add more if it's the condition is not good, some stress or water water quality is disturbed, you may want to increase the level. Okay, let's have a look at vitamin E. Vitamin E is a potent lipid-soluble antioxidant. It's found in higher concentration in immune cell compared to other cells in blood. So it is one of the most effective nutrients known to modulate immune function. So this is very, very interesting. Yeah? Vitamin C is a very potent antioxidant. So meaning to say antioxidant is very important in the modulation of immune function. By preventing lipid peroxidation and the associated cell membrane damage, vitamin E may assist in the maintenance of membrane integrity. Vitamin E acts as a second line of defense to prevent the proliferation of such chain reactions by scavenging fatty acid peroxy radicals that are produced during or before this chain reaction. You remember lipid peroxidation? Yeah. For example, feed contain fish oil. Fish oil is high in HUFA, highly unsaturated fatty acid. This highly unsaturated fatty acid, when it is exposed to atmospheric condition, it will be very, very easily to get oxidized. Oxidation of lipid will produce what do you call a toxic material. Yeah, this one especially will happen. This peroxy radical will be developed. So this thing we need antioxidant. In this case, vitamin E is a very good form of antioxidant. Okay, let's have a look at this study effects of dietary vitamin E on the growth performance, antioxidant status, and innate immune response in juvenile catfish, yellow catfish. I don't know if you have yellow catfish. I haven't seen any yellow catfish in Malaysia. We have a different species. This is from a study from China. Yeah, they developed 6 diets of different level of vitamin E, or known as alpha tocopherol. So and then they analyze the growth, weight gain, SGR, Fe, protein efficiency ratio, and it turned out that they were significantly influenced by this level, this different level. And the maximum of growth performance occur at this diet, 39.7 produce the best growth parameters, this one. Fish fed the diet containing 8.9, which is the lower. Vitamin E has lower HSI. You know HSI, right? Hepatosomatic index, the liver. We weigh the liver and then we calculate its weight against the body weight. So fish fed the basal diet had lower hematocrit and higher triglyceride concentration in serum than those fed the diets without vitamin E supplementation, meaning to say vitamin E influenced the hematology profile of the fish also. Moreover, fish fed the basal diet had lower activities of superoxide dismutase, SOD, and catalase in serum than those fed diet supplemented with vitamin E. Okay, fish fed the diet containing 39.7 milligram per kilogram vitamin E had higher lysozyme and respiratory burst activity in serum and the phagocytic index of head kidney. Remember head kidney? Then those fed the other diet. And the challenge test with Aeromonas hydrophila revealed that fish fed the basal diet had lower cumulative survival than those fed that diet supplemented with vitamin E. So in this experiment, the role of vitamin E is very, very significant. Improve the immune system of the yellow catfish significantly. I think most of the parameters were all significantly improved. Okay, continue with vitamin E. Hang on, yeah. I think this is done. Okay. Now we look at minerals. We have covered several examples or case study for vitamin C and then vitamin E. Now we look at the mineral. Selenium plays an important role in the maintenance of fish health, in particular fish immunity. Symptoms of selenium deficiency in fish include low glutathione peroxidase activity, and then reduced growth, impaired reproduction, anemia, exudative diathesis, muscular dystrophy, and increased mortality. You can Google what is, if you're not sure what does it means. Selenium is an integral part of the enzyme glutathione peroxidase, which protects cell membrane and tissues against oxidative damage. Selenium is required for normal growth, development, and flesh quality. It also stated here, symptoms of selenium deficiency in fish include low glutathione peroxidase activity, reduced growth, impaired reproduction, anemia. Sorry, this is a repetition of this one. Sorry, this is, you ignore this. However, excessive selenium can also cause a variety of toxic effects at the biochemical, cellular, organ, and system level. Sometimes vitamin E and selenium function synergistically in animal tissue to form important antioxidant defense system. You know selenium, right? We don't put that much in the diet, but even though we don't put much, the function is so significant. Even in our human diet also selenium plays an important role. I'm not sure here. In Malaysia, we have this so-called egg with selenium enriched, meaning the chicken, they will feed with selenium addition. So when they sell the market, this egg will carry more, will have more, a higher price. Selenium egg, something like that. So this is one way of marketing, of course, but what I'm trying to say, it is really proven that selenium is a mineral that is important for immune response of fish. Yeah, but of course, whatever nutrient, mineral, vitamins, or whatever essential nutrient, we need to determine the optimum level, because if not, then in excessive, if we put extra selenium, then it suppose we can put a toxic effects to the cell, to the system. So it's quite dangerous also. That's why there is a need for us to determine requirement, like the one in the lecture by Yomin just now. Yeah, the need for us to determine the optimum requirement, meaning to say, if you give your fish more than what they need, one, it will be a waste. It will increase cost. Why you want to put extra selenium, for example, extra vitamin E, if they don't need that high? That will increase your feed cost. Two, high excess level might have some negative effects to the health of your cultured animals. So as much as possible, we want to give the nutrient at the optimum level. If we give below the optimum, also no good, because later it won't be able to support the good growth or maybe become not so healthy. So that is the importance of giving nutrient at the optimum level. So we have to determine the optimum level of the essential nutrient. So that this is another topic of interest if you want to do your research. For example, a new species X that discovered or that is successfully bred in somewhere in Indonesia, you want to determine this requirement for whatever vitamin, mineral, or whatsoever nutrient. That is the purpose, because we don't want to give low or more than what they need for various reasons, for the cost, for the benefit of the species. Okay, this is also another study on mineral, which is on selenium also. In this study, they are using tilapia. Fish were divided into two groups. A control group fed a basal diet, and a treated group fed the selenium supplemented diet at 1 milligram per kilogram for 1 month, only 4 weeks. In the second phase, fish were divided into 4 groups. Yeah, group 1, the control. Group 2 was fed a controlled diet and exposed to cadmium. Group 3 was fed selenium supplemented diet, and group 4 was fed selenium and then exposed to cadmium. Yeah, exposed to a toxic condition lah. Okay, dietary selenium increased phagocytic, remember phagocytosis, and lysozyme activity in the second week, whereas phagocytic index and growth indices were increased in the 4th week, meaning it's proven that selenium added diet help fish in boosting immune system against suppressor. In this case, what is the suppressor? The cadmium toxicity. Suppressor, remember, doesn't have to be living, doesn't have to be pathogen. It can be non-living suppressor, environment, toxic, pollutant, all this can be a factor influencing reduced immune system of fish. IGF, I'm not sure, hang on here, what is the insulin growth factor was upregulated after 2 weeks, and TNF is tumor necrosis factor. IL, what is IL here? This is interleukins and catalase gene upregulated after 4 weeks, meaning at 2 weeks belum cukup lah maybe. So these two are can be detected after 4 weeks. Cadmium causes gill hyperplasia and degenerative changes in visceral organs, elevation in MDA, the melandialdehyde, and attenuation in glutathione, reduced activity. However, in G4 group, the histopathological alteration and antioxidant enzyme were elevated. So the conclusion, selenium improve growth, immunity, dan antioxidant power of this Oreochromis niloticus, and alleviate the pathological disorder induced by cadmium toxicity. Okay, let's have a look at probiotics. This is a very important topic also because it is what the industry is practicing for quite some time, and yeah, there are many development out of this introduction of probiotics since then. Probiotics are often defined as live microbial feed supplement, which beneficially affects the host animal by improving its intestinal microbial balance, and when administered in adequate amount, confer a health benefits on host. Nowadays, probiotics are also becoming an integral part of the aquaculture practices to obtain high production. The common probiotics that are used for aquaculture practices include Lactobacillus, Lactococcus, Leuconostoc, Enterococcus, Carnobacterium, Shawonella, Bacillus, Aeromonas, Vibrio, Enterobacter, Pseudomonas, Clostridium, Saccharomyces species. So many different, quite wide range of bacteria used as probiotic. Actually, it's not difficult to prepare probiotic. There are many ways to produce it in lab scale or at home also. The involvement of probiotics in nutrition, disease resistance, and other beneficial activities in fish has proven beyond any doubt, which is quite true. Among the numerous health benefits attributed to probiotics, modulation of immune system is one of the most commonly purported benefits of probiotics, and their potency to stimulate the systemic and local immunity. Dietary probiotic, either monospecies or multi-species, meaning you can use one type of bacteria or combination of bacteria, can eventually elevate phagocytic, lysozyme, complement respiratory burst, as well as expression of various cytokines in fish. Similarly, probiotic can stimulate the gut immune system of fish, which marked increase in the number of immunoglobulin cell and acidophilic granulocyte. Okay, this is an example of publication in Immunology and Medical Microbiology journal using rainbow trout. Enhancement of immune response and protection induced by probiotic lactic acid bacteria against furunculosis. Furunculosis is a common disease in rainbow trout. So they use Lactococcus and then Leuconostoc, Lactobacillus. Yeah, they administer using oral administration to fish 2 weeks at 10 to the power 6 of colony forming unit per gram of feed. In comparison to untreated control fish, the phagocytic activity of head kidney leukocyte and the alternative complement activity in serum was significantly greater in all probiotic groups on the 2nd week. So with the exception of Lactobacillus sake, superoxide and ion production was also significantly increased in probiotic group, meaning not all bacteria perform the same performance. Yeah, in this case, the Lactobacillus did not produce significant impact on the superoxide and ion. Analysis of lysozyme activity did not exhibit any significant difference in probiotic and control group. So in terms of lysozyme, no significant difference. The fish supplemented with probiotic exhibited survival rates ranging 97 to 100, whereas survival was quite low in the non-treated group, which is only 65%. That is huge difference for a company, for farmers, for business. This is quite significant difference. This result demonstrates that probiotic supplementation to fish can reduce the severity of furunculosis and suggests that this reduction may be associated with enhanced humoral and cellular immune response. Okay, I want to show you the video on probiotics.
With products such as the newest version of Sanolife Pro 2, Aquaculture's innovation team has managed to obtain the highest number of active good bacteria per US dollar invested. This portfolio was developed based on continuous research and development that started years ago when Aquaculture teamed up with researcher David Moriarty, who is still considered to be the true pioneer in the field of probiotic use in aquaculture. Thanks to his work, together with Aquaculture, there is now plenty of evidence that, when applied properly, the use of probiotics is an effective strategy for modern and sustainable aquaculture. During a recent encounter at a shrimp farm in Thailand, David Moriarty and Olivier Decamp, Aquaculture's shrimp farming product manager and probiotic lead, looked back on fruitful collaboration.
When you started 20 years ago, what was the main purpose for you to develop this product?
When I first came into Asia, it was Indonesia, Thailand, disease was becoming important. So we had selected bacillus that would be effective against certain vibrios that were causing a lot of problems. When I first started, I was really worried by the huge quantities of antibiotics being used, because that has an impact on human medicine. It can make bacteria that cause problems for humans resistant to antibiotics.
It's a message that people got when you explained to them the issue with antibiotics?
There was certainly a lot of support from the people, clinical microbiologists working with human medicine, because they could see the problems of antibiotic resistance being transferred from the use of antibiotics in agriculture for growing chickens, pigs, for example, but also in salmon farming. And then, of course, with shrimp farming, because there was a transfer through from the animals, ultimately, and it wasn't just a problem of residues, but of bacteria that were transferred during the processing, which could then interact with bacteria in the human gut and transfer resistance to antibiotics.
And when they were using antibiotics, was it to control which kind of pathogens in the case of shrimp farming?
In the case of shrimp farming, the most important one was vibriosis. Mostly the species was Vibrio havii.
At the time, you were doing everything alone?
Originally, I was working with a small company, but we were doing it on a very small scale. And we had to sell liquid cultures, and it wasn't sufficient. The farmers had to brew them. So the ideal was to get freeze-dried powders of concentrated bacillus. And I could do that only once we started working with Inve. During all the travels, I met Patrick Sorgos. He heard me give a talk at a conference. And he was really interested in what I was talking about with helping control disease in shrimp aquaculture. And he said, well, why don't you come through Belgium and see what we're doing? So I did that. And the long story cut very short is that they asked me if I would like to work with Inve. And for me, Inve was really a company with integrity. It had a lot of experience. It had a wide marketing. So I thought it was a good way to spread the news, what I was doing with probiotics on a bigger scale. Working with Inve has been really fun. It enabled us to do what I wanted to do for a long time. That the facilities that you had there in Belgium were really good for developing the products. And then for the packaging distribution, the facilities here in Thailand have been really good. So we're able to do a lot more than I was able to do with the smaller companies that I started working with. And of course, having people use the products is most important. And that's where working with INVE was really exciting.
The move towards the use of probiotics, the understanding of quality, the level of education of farmers now has improved so much that all the work you did over these years, I think it paid off.
It's now 5 years since I retired. I haven't been involved with this business. And so it's been really interesting to come back and see just with what enthusiasm you and the INVE team have been working. The work that the INVE team that you've trained, in fact, we previously trained together, the enthusiasm that they're spreading the message about why it's so important for farmers to use probiotics. I'm really pleased to see that.
What is good is these people are still with us. They were part of the key training we do all these years ago. They are still with us, and now they are transferring to new generations.
Today, INVE Aquaculture has a dedicated team of trained professionals who continue David's work to meet farmers throughout the world and explain the benefits of probiotics to run a healthier, cleaner, and more profitable aquaculture operation. You can find more information.
Okay, I think maybe most of you know about INVE Aquaculture, right? I think the lecturer must know INVE Aquaculture because this is a pioneer in aquaculture industry. They have many range of product, the enrichment product, Artemia, you know, the Celco enrichment, everything. INVE is the pioneer, and they engage all the academician like Patrick Sogilus and all that are from they have connection with INVE. So it is a very reputable company in aquaculture. So one of their research is dealing with probiotics. Okay, effects of, okay, now hang on here. Okay, done with probiotics. Now we look at prebiotics. What is prebiotics? It's not the bacteria, yeah, it's no longer the bacteria. Prebiotic is a non-digestible food ingredient that beneficially affect the host by selectively stimulating the growth and the activity of one or limited number of bacteria in the colon. It's like the food of the bacteria, the probiotics, you normally in the forms of normally undigestible fiber, something like that, that is so-called helping probiotic to grow with this. That's why it is called prebiotic. Prior to the probiotic to works, we put prebiotics. So there are food ingredients that are not digested by normal digestible enzymes, but which act by selectively stimulating the growth and or activity of beneficial bacteria in the gut and consequently improve the health of the host. Prebiotics are often naturally present in the diet ingredients or are administered as concentrated exogenous sources. So you can add it separately. They are non-digestible substances that are neither hydrolyzed nor absorbed in the digestive tract, but are selectively fermented by certain microorganisms in the digestive tract, providing benefits to the host by improving growth, nutrient digestion, immunity and disease resistance. Okay, this is a paper example of prebiotics. The importance of prebiotics in aquaculture as immunostimulant, in this case of two species, Parus aurata and also Dicentrus labrax. We'll not go through this, but this is among the published articles about the use of prebiotic, because unlike probiotic, prebiotic is comparatively new, so not so many available publication for us to refer. This is one of them. like what it says here, the road is still long, further studies are required, but the use of prebiotics individually or coupled together can open the doors to pioneering a new model of alternative dietary supplement. So meaning to say there is hope, but still many aspects need to be explored and studied, yeah, when it comes to prebiotics. Maybe students who have to do research for your master or even for your undergraduate thesis, you may want to consider study this prebiotics. Okay, effects of algal supplement. We have covered the vitamins, minerals, probiotics, prebiotics, and now algae. Algae, either the micro or macro, are rich source of nutrient. I think we have the expert here, yeah, Dr. Uun, I think, is the algae person. So it provides essential fatty acid, polysaccharide, mineral, vitamins, antioxidant, several bioactive compounds, including laminarin, fucoidan, and also beta-glucans as immunostimulant in several fish species. This is just example of dietary supplementation with omega-6 long-chain polyunsaturated fatty acid. Rich algae modulate zebrafish immune function and improve resistance to streptococcal infection. So in this paper, what they do, they use algae biomass they add to the commercial feed at 7.5 and 15%, providing this arachidonic acid for the weight, for the, what is WT? The microalga Dislabosphera incisa supplemented group, and 13.6 milligram per gram of DGLA is for this one, dihomo-linoleic acid. Sorry. And then the group unsupplemented group used as control. So they analyze this tissue RA and DGLA, then the level turned out to significantly increase in the liver, corresponding the macroalga supplementation level. And then importantly, fish fed with 50% of both WT and MUT supplemented feed shows significantly higher survival compared to the control. 78, 68 to 46 is quite huge, yeah. So the elevated expression of genes related to inflammatory and immune response was evident post-challenge. Collectively, the result of the current study demonstrate the potential of microalgae-derived dietary ARA and DGLA in improving immune competence and resistance to bacterial infection in zebrafish as a model organism. You know that zebrafish is normally, we don't culture it for food, but it's a model organism. So in this one, it is used to see whether the supplementation of algae containing this two long chain PUFA improve or not the immune system. It's proven that it is working in this zebrafish. Okay. This one is using seaweed. Seaweed as dietary supplement enhance the innate immune response of rabbit fish. Yeah. Polysaccharide of seaweed has been shown to stimulate non-specific host immunity and inhibit bacterial activity. So this carbohydrate also positively modulate gut health and potentiate fish digestive capacity. Hallmarks of a prebiotic categorization. You remember the topic prebiotics? So putting this seaweed supplement may perform as prebiotic. Additionally, seaweed beta-glucan, you know, beta-glucan is very, very good, what do you call, substance in stimulating our immune system. It is found in many types of plants, including seaweed. So they stimulate the immune system through the release of ROS, reactive oxygen species, and signaling proteins. Seaweed phenolic compounds may also wield a scavenging effect, reducing ROS formation in fish tissue. So red seaweed such as Glaciaria are rich in arachidonic acid, the precursor of the pro-inflammatory mediators prostaglandins, thromboxane, and leukotrienes. So yeah, this is just to share with you example of the use of seaweed as supplement to increase immune response. Okay, ini what is this? Oh, okay, this is actually a paper that I co-author with someone in Taiwan. You know, Taiwan is a very modern agriculture country. They have lots of lemon, oranges. They are very famous with agriculture product like pineapple. They have many, including this lemon. So with the excess of this agriculture product, lemon, they use lemon peel. The skin you can peel and then fermented and supplied as a supplementation in the diet. In this case, they are using Asian sea bass. So vessel diet supplemented with 0, obviously the control, 1, 3, and 5 of lemon peel, fermented lemon peel. These 4 experimental diet were fed to the therapeutic group of juvenile essence. It was only about 11 gram in recirculating rearing system for 4 weeks. Sorry, 8 weeks, 2 months. And then,
I'm sorry, Prof.
Yeah.
Your time is only 20 minutes.
20 minutes
left.
Okay.
Yeah.
All
Then
right.
we continue to Q&A session.
All right. Thank you for the reminder. Okay. In a circulating rearing system, so final weight gain, feed intake, survival was similar, meaning to say the addition of fermented lemon peel doesn't really have benefits on weight. Yeah. But plasma lysozyme was higher in fish fed with diet 3% than that in fish fed 5%. So meaning to say this particular plant supplement, the fermented lemon peel, doesn't have benefits on growth, but it somehow improved the intestinal health for Asian sea bass, and also may improve immune response in general, because this lysozyme showing the positive outcome. But putting more than 3 at this 5% suppress the immune response. So that's why we are determining different level, even though certain substances may have medicinal value, but the level included in the diet also important. In this case, it cannot be put too high. More than 3 could be detrimental to the fish. Okay. Okay, this is diagram just to summarize types of immunostimulant in fish. One is natural, the other is commercial. Meaning to say, like the video that I show from Biomin, you can buy your immunostimulant. Yeah, there are many different range according to the objective of your farm. You want to reduce the cost, you want to help the environment, what else? Many different criteria about the classification or group of additives. In this case, the natural one, you can extract your synthetic chemicals, microbial derivatives, and then natural, like what I did, I do this. I studied vitamin C, vitamin A on natural ingredients. Animal and plant extract. For example, you want to extract some bioactive compound from mushroom, for example, or any other plants, herbs. We will cover that during our lecture on Friday. And then polysaccharides also, hormone also is considered a natural immunostimulants. Okay, but I don't know much about hormone supplementation, because as much as possible, we don't want to like, you know, supplement our feed with hormone, because it, I don't know, somehow people will hear it something like sounds not very positive, right? People are avoiding taking hormone, because when your fish is supplemented with hormone, it will end up in your system. So we don't want that to happen. Okay, this is commercial. They have different types. Yeah, they're macro guard, immersion guard, aqua solo, macro guard, different types of commercial. Yeah. Okay, this is the effect of immunostimulant on fish. We can wrap up as these two categories. One is in vivo and the other one is effect in terms of in vitro. I'm sure you know what the meaning of in vitro and vivo. So in the in vivo effect, we can see that many research just now that I share with you able to increase survival or reduce mortality when challenged with pathogenic bacteria. Yeah. Anti-parasitic effects including reduced settlement of sea lice. Sea lice is quite common in salmon farming. So when we use immunostimulants, so we can do in vitro research. And then growth enhancement, you've seen that many supplements able to improve the growth of the fish. And then increase antibody production also. Some study prove that including immunostimulant in fish will increase antibody production. And of course, lysozyme is also another popular parameter that researchers like to analyze, maybe because of the straightforward methods or things like that. It's not very costly, easy to conduct, something like that. That is in vitro parameters. Meanwhile, that is the in vivo. The in vitro is more to the cellular, what do you call, parameters that we do outside the body of the animal. For example, macrophage activity, and then phagocytosis, free radical production, enzyme activity, production of cytokines, migration activity, lysozyme activity, and then cell proliferation and oxygen radicals. So I think I end my lecture there. This one is okay. This is only a video, so yeah, you can watch it later. You can see the address here in the YouTube. You can watch it later. So yeah, that's all for this topic. If you have questions.
Okay.
Oh, it's done. Okay. If there are any question, please raise your hand. Oh, there is no question yet. Maybe you can raise your hand if you have any question from this material. Okay.
Maybe you can write it in the chat as well. Okay, if not, probably
There's no question.
Still no question. Okay. Probably I will ask Prof Rosita.
Ya, silakan Bumi.
Yeah, okay. Prof, you know, when you have a research with vitamin C or E, or even combination of that, do you use like chemicals, vitamin, or vitamin based on the plant or local ingredient or something like that from.
We use the commercial vitamin available commercially, vitamin C, ascorbic acid, and also the other form. Yeah, it's not like I use natural from plants or anything. No, it's just vitamins for the feed ingredient.
I see.
Yeah.
So if for instance, some plant or herb or even other organism that had vitamin C in it, we can use that, or we have to, for instance, like determine first the content of this vitamin C, and then we put it. Do you think there will be got a different effect?
Yeah, there will be different effects because the vitamin we use is in pure form. It is vitamin C form. Meanwhile, if you use, let's say, I take some leaves that is known for vitamins high in vitamin C, but when I crush the leaves, the herbs, I make it powder. That is not in a pure form. We need to extract out vitamin C out of that particular plants. So the extract, the powder may have vitamin C, but it's not in pure form, meaning the content will be very low content compared to those I use. I'm using the pure vitamin C form. Unlike if you have plants, there are other components in it, right? There are several other components. So I normally use that kind of research also, Bu Umi, but then the objective is not like not focusing on the single content of the plant. For example, when I add seaweed powder inside my pellet, inside the diet, so the seaweed powder contain different types of mineral, a lot of minerals. They have antioxidant, but then some bioactive compound may not function the same as when I use the the pure form. It is not what do you call, not ready biologically available. Yeah, something like that. But I think you still can do that kind of research, but then it's with a different focus, not with the title, to supply vitamin C or something. No, because it is not in the pure form, meaning
in the pure form. Okay.
General supplementation, like effect of for example, whatever cumin or whatever.
I think as you mentioned before, in Thailand, they use like the peel, orange peel, the skin of the orange. I think they got also really high, relatively high vitamin C, I suppose, this skin of the orange. Don't you think so?
Yeah, I don't think they analyzed vitamin C in the orange peel. I don't think so. They don't analyze vitamin C, so we don't really know for sure what are the vitamin, what are the contents, or whether the vitamin is there or not. Hang on, where is the?
Okay, I was just recall that from a few years ago, I got like students. I don't know if Uun also remember these students. She also extracted vitamin C first and try to purify, even though probably not really good enough, since it depends on the new facilities or the equipment she used in the laboratory. So then based on that, then she tried to apply it in the feed and try to see whether the immune response are different by that or not. Because this plant, I can't remember exactly what is the plant, but it was containing, it was already some theories say that this plant contain vitamin C, I suppose. Yeah, so that's why. Yeah, probably. But yeah, now I know it will be give a different impact.
Yeah,
though.
definitely different because they are not the same form. For example, fish meal is very high in phosphorus, but we still add phosphorus in our diet if even we use fish meal. Why do you want to add phosphorus if fish meal is high in phosphorus?
Yeah.
The phosphorus that exists in fish meal is in a form that is not, what do you call, is not in soluble form.
Oh,
so
okay.
yeah, when you use it, so even though if you extract out, you will have the phosphorus, but if it's in that form, the animal may not be able to utilize it directly because of the form is in like hydroxy epithet that is not in soluble form. So this is the kind of thing, you know, we know that some ingredient, oh, very high in this one, but actually they may not biologically available to the animal if you don't extract it out, something like that.
Okay, thank you.
Yeah.
Is there any questions or raise hand already? I would love to discuss many things with Prof Rosita. Is there any questions, Febri? I don't see it in the chat. See it still.
Maybe
okay, then. Yeah, Prof.
Maybe I can answer your question from this paper yang orange field from the not Thailand. This is from Taiwan. So if you see here, this orange peel, what they want to utilize is it has in fiber, but this is already fermented. Yeah. Yeah, it's not a normal orange peel, but they already subject to fermentation.
Yeah, basically it's already extracted though. Usually.
Yeah, so that it will help the gut, I think, to be more efficient in digesting whatever material they use in the fish feed. So
I see.
Yeah, this is the one
yeah,
of
okay, collected from local and they
so for example here this lemon peel has the flavonoids, yeah,
yeah, that's right. Yes, that kind of
activity.
active
Not
substances.
so much on vitamin C, I guess, but maybe the fruit inside, yes, it has high vitamin C, the fruit itself, but this is the skin, the peel. So yeah, they are
yeah, flavonoid. Yes.
Yeah, flavonoids.
Okay. Because there's still no questions, probably I will ask these last questions before we if there's no questions, let's still have some 5 or 10 minutes more, Prof. This is probably a little bit out of the topic, but still related. I would like to know the how a fish or aquatic organism they sort of have a mechanism to purify the heavy metal. Do you recognize or maybe have some information regarding that, Prof?
You mean
I guess in some level aquatic organism got some mechanism to purify themselves from the heavy metal
I'm
pollutant.
not sure, honestly. I haven't really came across into that topic, how fish, but if you are to see the logic of the fish system, you know, all the what you call pembuangan ikan apa semua, they have to, I think, go through excretion, drink. I think that the process will be be what do you call involving all the normal process of excretion in the fish when it comes to heavy metals. But I know that there are several studies trying to see the heavy metals in our fish product. So meaning, of course, there are, I think, some amount that cannot be flushed out from the system. We know that liver is the organ to store all these impurities. So we don't really, actually, it's not good to eat liver, can?
Yeah, that's why.
Animal organ.
The organs that contain most of these heavy metal, probably.
So,
Yes,
I'm sorry if I can't really answer your question properly.
Yeah.
For this.
I remember that because when you show before the phagocytosis,
yeah,
do you think that they was it considered like heavy metal component as what strange?
I'm not sure. Honestly, I don't know if phagocytosis will be able to engulf, engulf the heavy metal also.
Heavy metal.
Yeah, I don't have any experience in this, but yeah, yeah, I think fish excretion maybe to discharge, right?
Yeah, that's right. Because there was a journal, I mean, research said that sometimes even got like or contain very high heavy metal, they're still alive. Yeah, they can still alive, but
maybe there are certain level that they can tolerate, can the bioaccumulation. Normally, like I said, bioaccumulation of metals or heavy metal is normally in the liver. I think in the muscle also, right? When we analyze the fish,
some of them are in the muscle.
yeah. So I'm not sure how fish really, apart from this excretory process, how they maybe some metabolic also what do you call process detoxification.
Yeah, detox. Yes.
Yeah. There must be some physiological processes involved, which I'm not that familiar to be honest.
Yeah. And even now, I think the most updated pollutant or issue is about microplastic, I suppose. Microplastic.
Yeah, true. That is a very huge issue on microplastic. I think not only in your place, in our place also, plastic is everywhere in our beach, right?
Yes, that's right.
Yeah, so it's a huge responsibility for the young people like the student to educate your family first, your sister, your mother, your auntie, not to pollute the sea, not to throw your rubbish, the plastic into the natural environment, because this is a global issues whereby we have one section called marine endangered animal. So normally there are several like dolphins stranded in our backyard, in our campus. Yeah, when we open the stomach, it's full of plastic, like 4 kilograms of plastic, and it died after that. So it's very sad. Yeah, it's very sad, Bumi.
Yes, yeah. It happens in Indonesian water as well.
Yeah.
I think that kind of problem. Yes. And I believe that microplastic contented in their body will be also will be able to
reach us
impact. Physiologically or something like that.
So
okay. I think since there's no questions, probably
everyone is
Febri.
Yeah.
Febri, what do you think? I think Prof Rosita can have rest now.
We still have 12 minutes left.
You can ask any question, or you want to share something. maybe we can, yeah, still use this whatever time left to, yeah.
Prof, there's a question for Prof Sri Andayani in the chat.
Yeah,
what is the
I
difference between immunity in crab, shrimp, and clam?
In general, they are the same because we only differentiate to what science know now is between the vertebrate and invertebrate. If you remember the lecture, the early lecture, I differentiate between the vertebrate, which is the fish, and then invertebrate, which include the crab, shrimp, or even clams. I know clam is understudied animal when it comes to fish, what do you call, immune response. So most available, what do you call, available publication are on other species apart from, what do you call, clams. So in general, they are supposed to be the same under the what is it, invertebrate.
Yeah.
They have only non-specific lah. Mainly, the mediated by hemocyte, similar to the shrimp.
Yeah. I think Professor Sri Andayani is also a physiologist.
Oh, okay, maybe.
No different, no different immunity to shrimp, crab, and clam. No different.
Yeah, I think so. They are the same group of invertebrate.
Okay.
Yeah. Oh, okay. Febri, there's another questions.
Bumi, what is your expertise?
Mine?
Yeah.
Yeah. Mine is in especially in more in environmental.
You are not aquaculture group. You are looking for environmental like that.
Yes. Yeah. Basically, I'm from. My topic is about environmental science, environmental biology. Prof Rosita. So yeah, we sort of a little bit interested in something like what is the pollution or environmental disorder or something like that in the fish or in the physiological effect in organism and so on. So we try to understand that, and yeah, hopefully that this will help in manage the biota. If the environmental is healthy, I suppose the biota is also healthy as well.
Okay, interesting. Oh, ada satu soalan Gian, betul ya, Gian Surya Hartawan. Can we collaborate mineral vitamin with probiotic? Yeah, you can. But when you want to design your experiment, you have to be sure what is actually the purpose of the experiment. Do you want to determine the effect of vitamin? Or do you want to determine the effect of probiotic? For example, if you want to determine the effect of probiotic, maybe vitamin is not becoming the factor there, meaning to say you will supply vitamins in all diets at the same level, for example, and only probiotic becoming a factor. Or if you want to see the interaction, you can do vitamin and probiotics at different levels, for example, to see their interaction with and without, or low or high. Definitely you can do, but yeah, it's a different approach, different experimental design. For example, if you do have two different factor, it become, the way you analyze also become different, maybe two-way ANOVA or something like that, because of the purpose of your experiment. And of course, you can put probiotic together with the mineral and vitamin. There's no problem with that. Okay.
Okay. There was another one.
Yeah, last question for Genesia.
This one, Genesia Gerin Lucia. Hang on, yeah. As many ways to increase. To ask about which one is more efficient to increase the immunity of vertebrate. There is no right answer or wrong. I would say, yeah, I cannot say vaccine is better than immunostimulant. It's, I think, case by case, you know, different on what types of vaccine or what types of immunostimulant in what species, you know, in what dose. So I cannot answer either vaccine or immunostimulant. Yeah. This is very dependent on many factors, whether the vaccine is, you know, very good, suitable for this animal, or immunostimulant, what kind of immunostimulant. So depends. There is no specific answer that I would say vaccine is better than immunostimulant or the other way around. So depends on the target animal also, and also the type of vaccine, the stability, the effectiveness, and of course, the same thing goes to the immunostimulant also.
Yeah. But for sure, you don't vaccine the invertebrate, yeah, bro? It will be waste of time. Yeah.
If you ask for human vaccine or immunostimulant,
yeah,
is there
vaccine first.
Anti-vaccine here? It's also issues, yeah, during pandemic, anti-vaccine or, but yeah, we try to respect everyone. We know why certain people refuse to take vaccine for variety of reason, kan? So,
yeah, that's right.
Yeah, initially I also some you know, feeling a bit a bit scared.
Yeah, scared actually because of scared, Prof.
Yeah, true. So after reading different kind of reports and you know, experience from the country that already vaccinated, we feel more confidence and decided that actually the one afraid of vaccine, or the one is want to get vaccinated, the reason is the same actually. They are afraid of dying. Kita takut mati vaksin ataupun takut mati disebabkan tidak ada vaksin. So sama saja alasannya.
Yeah, betul. Yeah, especially the person who has comorbid, you know, comorbid.
Yeah. Those with underlying issue like blood, diabetes.
Yes, that's right. So they are really afraid of vaccine, vaccinated. Yeah, Prof, I heard that this COVID-19 can affect the animal.
Yeah, I heard also, but I don't know how true. Last time they said cat also can become, you know, carrier to this COVID-19. So I have several cats at home. So I was scared at one point, but I don't know if it's true or not. Yeah, more research is needed, I think. It's interesting, right, to see whether or not it can be, it can infect other animal also.
Yes. Okay, February, I think the time is almost up.
Yeah. Okay. Thank you, Prof, from the Dietary for sharing. It's really interesting. And also, I hope all of the students can take the advantage of your sharing. Thank you, and we will see you in the next day, tomorrow.
Yeah, sure. Yep. Tomorrow will be on, what is the topic? Oh, I think it's Industrial Revolution. It's a very interesting topic also, which is very related to our situation now, always using IT. So this is the topic using Industrial Revolution, like IT-based thing in aquaculture. So yeah, looking forward for our sharing tomorrow. Yeah.
We will see you again. I will see you again, Professor.
Yeah. With Umi, with Miss Umi. Umi. Sekali lagi, Prof. Thank you very much. And also to participant, thank you for your attention. Should we take a photo? Pak Dailami?
Yeah. Boleh. Take photo every day.
Yes, of
Okay.
course.
Please turn on your camera. So the participant, please turn on your camera.
Prof, would you please close your
I
share?
need to stop sharing. No wonder I cannot see our photo. Hang on, with our tudung.
Yes.
And we put our glasses. I cannot see.
Yes. Okay.
Okay, I will call.
For all the participants, would you mind turn on your camera, please? We will take a photo for a while.
Okay, they say. Okay. 1, 2, 3. Once again. 1, 2, 3. Okay, thank you.
Thank you so much. Thank you.
Thank you, Pak Dai.
Okay. Foto dulu bersama.
Ibu
Sudah,
Febriani.
moving for picture together.
Yeah,
Sudah.
sorry.
Okay.
Foto
Thank
lagi
you,
kita, Bu.
Prof.
Iya,
Yeah.
jamnya ini. Maaf, jamnya. Ada report jamnya.
Ada.
Kita ulangi lagi ya, satu kali
Kita
lagi.
ulang ya, Bu ya.
Bun belum ikut tadi.
Oh, belum ikut.
Jamnya gitu ya. Jam 12 ya.
Iya.
Oke. 1, 2, 3. Lagi. Senyum. 1, 2, 3. Baik. Terima kasih semuanya.
Iya.
Baik, terima kasih.
Sampai ke Mbak
Have
besok.
a nice
Prof Yani,
rest,
terima kasih.
Prof Rosita.
Kak Febri,
Bye.
Omi.
Thank you, Prof
Teman
Rosita.
Komita, terima kasih.
Terima kasih.
Waalaikumsalam.
Ya, sampai jumpa besok.
Ya.
Yes.